So thanks to Ohm and Joss for organizing. I have to go to many meetings every year, and some I want to go to, most I don't want to go to. But over the decades, IPC is always my favorite meeting to go to, and no exception here. So what I'm going to do, Mike, better? So what I'll do today is talk about some of the work that we've been doing, looking specifically at how to create climate resilient crops. So I'm just the mouthpiece here. Actually, a lot of these ideas came from Li Juan, who's at Nanjing University. So the nanotechnology research program at the experiment station began in about 2008 or 2009. My training, my PhD is in environmental toxicology. So when I started hearing about engineered nanomaterials in consumer products, specifically in agricultural products, I kind of viewed them as the next emerging contaminants. So what we did was we set up this program on nanotoxicology, addressing all of the questions we felt you would want to answer if you were concerned about the toxicity of these engineered nanomaterials being in pesticides, fertilizers, consumer products, biosolids. And what we noticed over 4 or 5 or 6 years of working was that not only were there certain instances where these nanomaterials were not toxic, but there were actually scenarios under which you could convey significant benefit to plants. So then that's when this whole second area of research opened up, nano-enabled agriculture, and 5 of our postdocs are here presenting either in poster or presentation platform on a lot of that work. But retrospectively, after looking at a lot of that work, what we noticed and what I noticed as a toxicologist is that the difference between dose that caused toxicity and the dose that caused benefit was actually really, really narrow. And fundamentally, a lot of it was related to ROS, reactive oxygen species. So what I'm going to talk about today is some of the work that we've done kind of looking at our research over the last 10 years or so retrospectively and kind of and projecting forward where we want to go in terms of climate resilience. So ROS, I think most people are familiar with. These are reactive oxygen species. And where I'll finish up is the flip of that, where you can actually ahead of time induce this ROS signaling pathway to stimulate plant defense before the stress has occurred. And when you start thinking about climate resilience, that's when you really see this could be important. So the left pathway is recovery, which is what we'll talk about. But this right pathway is preventative, which I think is a little bit more exciting. But again, so this is where we started working on this in plant disease systems. We began in 2014. Our first paper we published in 2016. And we've talked about this before, the fact that you're losing 20% of most crops to plant disease. And we chose to focus on fungal pathogens because they are particularly difficult to deal with. We don't fumigate soils anymore. So it's really difficult to get rid of the pathogens. In the US, we're spending $600 million a year on fungicides, and we still lose $200 million worth of food to these systems. So we're focusing on these nanoscale micronutrients. because they're really important to all of these plant secondary metabolite defensive pathways, and their availability is critical to the plant's ability to respond. And the availability is a really important issue because when you add these in soil, when you add these foliarly to plants, the availability is often quite low, and we've talked about that before. So that's why we're using nanotechnology, because we can boost that availability and, in theory, stimulate these systems. So what we like to point out is, although we're working in disease systems, we're not targeting the pathogen directly. We're targeting the plant's ability to respond to that pathogen either before infection or after. So this is our first study. There's a lot of data here, so I'll just walk you through it quickly. But this is the kind of work we started doing. Agriculture in New England is largely specialty crop based, so a lot of farmers will start seedlings in the greenhouse and then move them to the field. So that's the system we chose to work with. We used eggplant and tomato because those are good disease systems we had with Fusarium, a fungal pathogen. So we did a foliar application of our nanoscale micronutrients, but you always have to use your bulk and salt comparisons, as was mentioned yesterday. You never want to go nano unless you have to. And so we do a foliar application to the seedlings, and then the plants go into the field for a full life cycle study. So the short of the long here is this is the eggplant data. The green bars are copper. The center green is the nanoscale treatment. And what you see is we've got greater yield with the nanoscale treatment of the seedling. You got significantly less disease being evident, and you get significantly greater copper in the root system, which is where we want to stimulate these pathways. So we're basically alleviating the stress that's caused by this pathogen. I'm not showing you that data directly, but we're alleviating that stress, and that's allowing that increased yield. The other thing that you always want to keep in mind, and we talked about this yesterday too, that these strategies need to be both simple and affordable. So in this case, we spent about $44 on nanoscale copper oxide for a field study, and we increased our food production, which is the way I think about it, significantly. But economically, you're looking at about an increased yield of about $10,000 per acre with this simple foliar application of a nanoscale nutrient, again, to stimulate these pathways. So this is the paper where we actually kind of demonstrated this conclusively. This is a paper we published in Nature Nanotechnology at the end of 2020. It's part of this NSF center I'm in, where we're really focused on the chemistry of the material and how controlling that chemistry can lead to different impacts on the plant. But here what we showed was the different forms of nanoscale copper that we used, again in comparison to the conventional material, against soybean and a fusarium that causes soybean sudden death syndrome, SDS. So the disease is reducing biomass in all of our parameters that we're interested in by 60 to 70%. But again, our foliar application of specifically nanoscale copper is able to alleviate much of that damage. All of the disease is channeled through all the disease damage is channeled through oxidative stress pathways, and it's our ability to alleviate those to alleviate the stress from those pathways that's actually mediating the benefit we're seeing. What was interesting was we were actually able to correlate a lot of the effect directly to the properties of the different types of copper, whether it was a nanosheet or a sphere, how quickly it dissolved. And we were able to describe that computationally, which is why we ended up in Nature Nanotechnology. But the key point is it's these oxidative stress pathways that we're alleviating. Another material that we worked a lot with is sulfur. You saw Sudhir talk a little bit about our work with sulfur and toxic metals. The focus of this part of the project was sulfur as nanoscale sulfur as a novel form of nutrition. We had both greenhouse and field studies here. I'll just go through this quickly, but again, a couple of things to always point out. You always want to have your conventional controls here to make sure that, again, your nanoscale form is necessary. This was a study with tomato and another form of Fusarium, a fungal pathogen. In the greenhouse study, the postdoc, who's now a scientist at Case, measured all sorts of endpoints in terms of what we were looking at, including time-dependent analysis of gene expression and metabolomics directly related to stress pathways. But similar type of data to what we looked at before. The Fusarium is going to reduce biomass. This is a greenhouse study, so it's just biomass, by 87%. But our forms of nanoscale sulfur are able to alleviate much of that damage. And there were some other really interesting things we noted. You know, when the sulfur was in nanoscale form. It was actually activating different uptake pathways and feeding directly into glutathione, which is a key part of our stress alleviation. And this is just showing you some of the time-dependent metabolomics. And I'm getting a message here about closing all rooms. So I guess I will just get rid of that. You guys probably can't see it. Okay. But again, it was the ability to overcome stress was critical here, and it was a nanoscale sulfur-specific phenomena, and actually the coating made a big difference as well. But one thing to point out again is here we are doing a field study where it was the same exact experimental design, except none of the endpoints other than yield, because that's what you ultimately want, and nutritional quality of the yield. So nutritional quality was enhanced. We were seeing a biofortification. I don't have that written here, but that's a separate part of our project. But again, a $33 investment in nanoscale copper or nanoscale sulfur, and look at the increases in yield we're getting: 4,900 kilograms of tomatoes per acre to almost 12,000. So that's a significant boost in food production just by adding this in this case nanoscale sulfur to the soil. This was a soil amendment. So obviously, all of the stresses are not just biotic. This strategy works with abiotic stresses as well. This is a paper we actually published all the way back in 2019. Chris Dept did this one, where he was exerting drought stress on sorghum in a greenhouse study. And what he noticed was, what he observed was that you could add nanoscale zinc oxide to the soil at relatively low concentrations, and you could significantly increase the ability of sorghum to withstand this drought stress. So in this case, the D is drought, not disease, but you can see yield and all these other parameters drop off when the sorghum is drought stressed. But by just making this simple amendment of nanoscale zinc, and again, it's specifically nanoscale zinc, the conventional forms don't work, you see a boost in all of these parameters. And guess what? Sure enough, it's all mediated through the quenching of these reactive oxygen species that are produced as a function of drought stress. So we're modulating the plant's ability to respond to stress by supplying these extra nutrients, micronutrients. So not just drought stress, it works with salinity stress. This is back to our center. And in this case, we're looking at cerium, which is interesting because cerium is actually not a plant nutrient, but it's a known antioxidant, and it worked exactly the same way. This was work we did with cotton. It's one of the few studies I've done with a non-food crop, but the center had money from the cotton board in California, and when you get money from the cotton board, you work with cotton. But what we did was actually a seed treatment with a coated form of nanoscale ceria, again with all the appropriate controls. And sure enough, when you take those primed seeds and you expose them to drought stress, again you see increased biomass, increased vigor. When you actually measure the ROS, they're reduced. And when you look at some of the genes that are expressed, again, it's all around these stress response pathways. So this seems to be across materials and across plant species. You can really facilitate a plant's ability to recover from a stress that's occurring, biotically or abiotically. The last one I'll talk about in terms of this reactive pathway, recovery pathway, is nitrogen fixation. Specifically, we're looking at biological nitrogen fixation. These are just 3 studies we have recently. I'll focus on one in a minute. But you can't talk about the inefficiency of agriculture without talking about nitrogen, right? It's kind of the elephant in the room, nitrogen and phosphorus at least. So we're focused on biological nitrogen fixation, which is these symbiotic relationships for the most part, where bacteria do the nitrogen fixation for the plants, and then the bacteria also get significant benefit. The key point for us is that 2 things. One, in the environment, in agricultural systems, biological nitrogen fixation occurs at relatively low rates because we're supplying so much synthetic nitrogen. So the other issue is that abiotic stress is a real problem here. These nitrogen fixing systems are very sensitive. So in our case, we want to see how we can work with molybdenum specifically, because that's the coenzyme metal center for nitrogenase and a couple of other key enzymes. So can we supply that nutrient in nanoscale form to do this same thing, to convey some stress resilience to this process? And guess what? The answer is yes. So this was a study we published in ACS Nano last year, at the end of last year, looking at different forms of nanoscale molybdenum compared to the conventional controls. In this case, it's a soil amendment because that's where this process is occurring. Time-dependent harvest, measuring all sorts of endpoints. But the key point is when you're supplying this nanoscale molybdenum, you're able to boost this system. You're able to make it more resilient to stress that's occurring just by natural ROS accumulation. So how's it being mediated? Actually, the mechanisms vary over the life cycle of the plant, where you're seeing at the reproductive stages, you're seeing a longer release of the molybdenum, a slower release, which is stimulating nitrogen fixation. But earlier on, it's all about the ROS capture. So the take home here is you're getting this nanoenzyme function, and you're able to maintain your ability here. So I will go really quickly. We're doing a lot of work in this area now, including some green biosynthesis, which Mila talked about yesterday. Just to finish up quickly with the other part of this, can you actually prepare plants for stress that's going to occur later? So this is stress training. We do it in the form of seed priming and leaf priming. This is a paper we published also in ACS Nano. The idea is to treat the seeds with a low level of something, in this case silver nanoparticles, which stimulates all of these defense pathways first. And then later you're exposing it to a stress. In this case, we were exposing it to biotic stress. This is rice, later exposed to blast fungus or cold stress. But these stresses are occurring literally 30 days after the priming has happened. 30 days after you've exposed the seeds to those treatments that are going to stimulate these defensive pathways. And as noted, it works. So this is the amount of disease on the leaves in the control. And these are all the different combinations that were stress primed. So you're seeing significantly reduced disease when infection occurs a month later. And then there's the cold stress data. This is another study we were published in EST where we did combinations of stress, whether it was drought plus salinity or there were a couple of combinations. But the point being, even when you had combinations of stresses, if you were working with seeds that were pre-adapted to deal with those stresses, they did better. And I'll skip this one as well because I know I'm about out of time. But the other thing that we've noted, we haven't published this yet, is we've collected seeds from some of these primed plants and done the F1 generation. And there's actually some stress memory that seems to happen. Those F1 plants, those F1 seeds and seedlings are also still more resilient to different apiotic stresses. So again, the idea here is can we increase the climate resilience of agriculture? I think the answer is yes, and I will stop there and answer questions.
Thank you very much, Jason. Okay, student question first, please. We can't move until a student has asked a question. Come on, students. There must be loads of questions we can ask this eminent professor. Hands up. Okay, let's have a question from over there then. Student. Yeah, come on, student first. Come on, we've got questions about the metals, the size of the nanoparticles, what happens in the soil. Students, come on. That's it, there's one there smiling. Quick, what's your question? Come on.
Please. She won't give up, I'm telling you.
Right, okay, got questions here. What about the accumulation of the nanoparticles in the soil? Come on, girl power, you can do this. Yeah. Jason, what about accumulation of nanoparticles in the soil? Come on.
Liz, there is a question there from student.
Oh, we got one over there. Come on then, Erin.
All right, Erin, hit me with it.
Okay. Correct me if I'm wrong here. It sounded like you said that for the copper nanomaterials, you did a foliar application, but for the others, they were soil applications. Why was that?
Great question. So the type of application we do actually depends on the process we're trying to stimulate. Mm-hmm. We try and stay out of the soil as much as we can because soil, and my PhD advisor was a soil chemist, so he's turning over in his grave, but soil is a giant black box. So you want to stay out of the soil as much as possible. Baoshen's probably mad at me too. But by doing a foliar application, you can add far less material and you get much greater control over what is happening where. But certain instances you do need to go into the soil. Growers in the US are adding sulfur to soil for sulfur deficiencies. Nitrogen fixation has to be in the soil. But I would say probably 90% of our work in this area and all of our other projects, we're trying to do foliar application. And it's a function of trying to use the lowest amount of material possible to get the greatest impact. And I think there was a question over here about the accumulation of nanoparticles in tissues.
Okay, ask him about the accumulation of nanoparticles.
Is there any negative result during this when the nanoparticles is applied in the soil?
Yes, so there certainly can be. So the interesting thing about this whole strategy is it's all a function of dose. I've killed as many plants with nanoscale copper as I've helped. So it's a function of dose, using the right amount of material at the right time by the right application route. For every one way you can use nanotechnology for benefit in agriculture, there's probably 10 ways you can do it negatively or have a negative impact. So you really need to understand the mechanism of your material and the process you're trying to enhance.
Sir, hello, sir. Sir,
Yes, in the back.
sir, you said that the ROS generating nanoparticle help to alleviate abiotic stress. How actually it works?
So reactive oxygen species are an evolved intracellular message. So when any organism is under stress, so let's just say I was super nervous to give this presentation. I would be under stress, reactive oxygen species would be generated, and those ROS are a signal that there's an external stress affecting me. So all we're trying to do is take advantage of that process, that natural evolved process in plant cells, and stimulate it at the right time, so that all of my defense pathways are upregulated. So if I walked off the stage and fell down the steps, I would be better able to handle that stress that occurred later, which now I'm really nervous about. So,
Can I?
So, a wonderful presentation covering very large applications of the nanomaterials in elevating stress conditions. I just a query. I want to know what is the key mechanism which are involved in using the zinc nanoparticles for in the evaluation of the drought stress, because a lot of crop yields are affected by the drought stress in the South Asian countries and globally.
yeah, so zinc's an interesting one. It's not the material we've used the most, but I think it's interesting because a lot of soils, as you say, are zinc deficient. And so you've got this combination of zinc deficiency and drought. But in this specific study, it was the accumulation of nanoscale zinc and then the implanted dissolution that was able to basically directly quench the reactive oxygen species that were being generated by the drought stress. If I had that to do over again, I would have done a foliar application because I think it would work the same way with 10 times less material. But again, it's all through a nanoscale zinc-specific stimulation of these ROS pathways.
Okay, thank you.
Can I?
Ask the chairs. I don't know. Okay, thank you.
Thank you very much. We have a slight change in the program. Our online presenter is having difficulties connecting, so we are going to go with Dr. Marta Marmirari from University of Parma. The title of their presentation is Green Synthesis of Copper Oxide Nanoparticles from Macroalgae Ulva lactuca and Gracilaria verrucosa. Thank you very much, Dr. Marmirari. It's all yours.
So that should be no problem. It will be the next presentation. No? That should be fine. Yeah, that should be perfect. Fine.
Can I start? Okay. Green synthesis of copper nanoparticles from macroalgae Ulva lactuca and Gracilaria verrucosa. Good morning, everybody. Thank you for allowing me to give this presentation. Thank you to the guest and to the chairman. And we start with the aim of the project. This study focuses on green synthesis of copper nanoparticles from algae, Ulva lactuca and Gracilaria verrucosa. The biosynthesized copper nanoparticle was successfully characterized using many strategies, including biochemical analysis. The study demonstrates that it is possible to obtain useful products from these macroalgae through a green synthesis approach, and that they should be considered not just as a cause of environmental and economic damage, but also as a potential source of income. This is Italy, as everybody knows, and we have in the north of the Adriatic Sea the Sacca di Goro, where there is the cultivation of clams, and the blooming of Ulva lactuca and Gracilaria verrucosa, which are two macroalgae which are rich in polyphenols and soluble sugars. To go to the green synthesis of copper nanoparticle, we harvested the macroalgae and we dry them, and we obtain with a dry biomass ethanol ratio of 1:30 on a hot plate under magnetic steering the extract of the macroalgae. At the end, sonication was performed to maximize dispersion. The extract was collected using centrifugation and stored at 4 degrees before nanoparticle synthesis. Copper oxide nanoparticle synthesis was carried out using copper acetate solution at 1%, and synthesis was performed by a microwave-assisted method. The synthesis of raw copper nanoparticle was completed when the color of the solution changed from light blue to greenish, deep greenish blue. Continue the green synthesis. The isolation of raw nanoparticles was performed using centrifugation. The temperature was then raised from 4 degrees to 110 degrees and maintained for 2 hours to remove final traces of water. Raw copper nanoparticles were cooked in a ventilated oven at 1200 degrees for 10 hours to obtain structured copper oxide nanoparticles. As you can see in the image, at the bottom of the Falcon tube there is a brown sediment. That is our raw nanoparticles. We characterize the nanoparticle with physicochemical analysis and biochemical analysis of different nature, like XRD, FTIR, transmission electron microscopy with X-ray detection, zeta potential and dissolution, the measure of the PPH, microbiological analysis, flow cytometry on yeast and Candida albicans. These are the physicochemical analysis results. For the XRD, we can say that according to the inorganic crystal structure database, the nanoparticles are composed of tenorite, which is basically copper oxide, and the dimension could be estimated within the range of 30-40 nanometers for both nanoparticles deriving from red and green algae. The broad peak in the region for the FTIR, the broad peak in the region of 3,500 to 1,000 can be the result of multiple absorptions such as those due to OH, NH and CH stretching. As you can see, we have a green signal and a red signal. The red signal are the red algae, and the green signal are the green algae, and they are basically superimposed. So the coating of the two of the nanoparticles derived from the two macroalgae is the same, and it presents OH, NH, and CH stretching, a peak that could be abundant in polyphenols that may be attributed to aromatic ring stretching, and another peak at 1405 could be due to bending of OH bonds. The peak at 787 centimeter at the -1 could be the absorption of another aromatic ring. For the transmission electron microscope and the X-ray detection, the aggregation dimension of the, and the size and shape of both types of nanoparticles were quite similar, most of them being around some tenths of nanometer, with an ovoidal round shape. The composition is copper, oxygen, and carbon. The zeta potential, the DLS, and the dissolution estimate for nanoparticle average size. We can say that the zeta potential for the green macroalgae nanoparticle is minus 20.83 millivolts, and for the red algae minus 31.66 millivolt. The dimension for the dynamic light scattering for green algae is 65.9 0.68 nanometers, and for the red algae is 110,000.73 nanometer. Average nanoparticle dissolution had been estimated as 0.1-0.15% in 20 days, which is consistent with previous analysis performed on copper oxide nanoparticles. For the biochemical analysis, we start with the Ross scavenging capacity measured with the DPPH assay. We have a standard, which are commercial copper oxide nanoparticle, the GH, which are copper oxide nanoparticles synthesized from green algae, RA, which is copper oxide nanoparticles synthesized from red algae, and DPPH, which is the untreated solution or the control. No significant difference between the control, the standard copper oxide nanoparticle, and the copper oxide nanoparticle derived from algae was observed, according to the student t-test. We continue the biological assay with the viability test of serial dilution of microorganisms treated in liquid media with copper oxide nanoparticles. 10 milliliters of cell culture was plated on agar media after 24 hours of incubation with the various nanoparticles. Copper oxide nanoparticles from green and red macroalgae behave in a similar way in inhibiting growth in the microorganism in liquid media where the viability is larger. They inhibit mostly the growth of Candida albicans, Saccharomyces cerevisiae, Escherichia coli, and Bacillus subtilis. They don't inhibit Staphylococcus aureus. On solid media, the nanoparticles synthesized from macroalgae did not inhibit any of the microorganisms except for Escherichia coli, which was inhibited by the nanoparticles from red algae alone at a concentration of 500 micrograms per liter. This observation is confirmed by the fact that there is a reduction in respect to the control at 2,450 micrograms per liter. When the microorganisms were treated with nanoparticles from green or red algae, there was decrease in growth of at least 20% compared to the control, except for Candida albicans, which grew unchanged in this environment. This data, as in the case of Saccharomyces cerevisiae, largely depend on the particle behavior in terms of stability, aggregation, and subsequent bioavailability. Flow cytometry distribution diagrams of flow cytometry with propidioiodide signal for cell mortality of Candida albicans and Saccharomyces cerevisiae treated with 0 or 250 micrograms per liter of either commercial copper oxide nanoparticle and commercial oxide nanoparticle synthesized from green and red algae. As an internal standard, a sample with induced 50% cell mortality has been included. There are no significant changes in propidioiodine signal, hence in mortality, between the different types of nanoparticles and in the two different organisms. Concluding, we can say that macroalgae, especially Ulva lactuca and Gracilaria verrucosa, are considered as invading species and noxious species by clam fishermen in the northern Adriatic Sea in Italy. Macroalgae cause problems for the environment and are detrimental for tourism in the area. We have demonstrated that it is possible with green synthesis method to obtain copper oxide nanoparticles of diameter between 40 and 70 nanometer from biomass of this species. These nanoparticles are valuable because of their antimicrobial potential. Thus, a marine nuisance can be converted to a useful material, which may be exploited for human well-being. This is the publication on nanomaterials of this year, if somebody wants to read the detail of the process of producing copper oxide nanoparticles and so on and so forth, and the analysis. And I want to acknowledge the National Recovery and Resilience Plan for Italy for funding the Ecosystem for Sustainable Transition in Emilia-Romagna project called Ecosystem, and this work has benefited from the equipment and framework of the CompHub initiative funded by the Department of Excellence program of the Italian Ministry for Education, University and Research in starting 2018, finishing next year. Thank you for your attention, and I'm open for questions.
Thank you very much. So questions. We are ready to have few questions.
From students?
From students first. Yeah.
If you haven't got one for this, you're going to need a question for the next talk. I'm giving you some warning to get a question ready. Any questions?
It goes one, two. Okay.
Hi, Marta.
Hello, Jason.
Really interesting project. So I'm wondering if it's with the antimicrobial goal that you have, is it possible to synthesize multiple nanoparticles at the same time? For example, to start with copper, zinc, and sulfur all on the same matrix, and then I know the conditions for each particle to produce each particle are slightly different, but I'm wondering if you could actually get a kind of multifunctional material at the end.
We try to synthesize together with copper silver nanoparticles, and the method with ethanol dissolution of the algae content doesn't work. We don't get nanoparticles. So we are trying another method with water instead of ethanol to synthesize both silver and copper oxide nanoparticle at the same time. So probably we will manage. We just collected the algae 2 weeks ago, and we are drying them just right now and starting the procedure next week. And we will see that if we use water instead of ethanol, maybe we can reach the combination of the two types of nanoparticles.
So one synthesis and two particles, right? Yeah,
Yes.
I don't think anybody's ever done that before, so that would be really interesting to see if that's possible.
That's what we wanted to do.
Yeah, nice.
When Milica was with us, we tried different methods, and the ethanol one seemed better. But at the end, with silver, it failed.
Right, thank you very much. Any other questions?
I got one here. Thank you for your talk, Marta. The question is, you right in this slide, your green synthesized copper nanoparticles have higher bioavailability. I'm assuming you have biomolecules coated on your particles. If that's the case, can you identify what type of biomolecules on the surface of your green synthesized nanoparticles?
We haven't characterized the molecules that are doing the binding of the nanoparticles. It's one thing that we wanted to do, but we never had time. So that is still an open question also for us. Thank you for reminding me that I have to do it.
Thank you.
All right, thank you very much. So if there are other questions, you may reach out to Dr. Marmarelli later. We are going to move on with our next speaker.
As Anne passes it so she can watch me pronounce the name. Okay. We have an online presentation from Perisetti Pahada Saradi. Thank you. Plants fabricate nanomaterials as an economically ideal channel to defend heavy metal stress. Okay.
So you can mute yourself. I mean, unmute yourself. Okay. Are you audible, sir? Sir, can you hear us?
Increase the volume.
Sir, can you say something?
Sir, are we audible?
Yeah, you are audible. Am I audible?
Yes, sir, you are audible. You can share your screen and ready for the presentation, sir.
Yes, sure. Is my presentation all right?
Yes,
are
sir.
you able to?
Yes, sir. We are able to see your presentation.
Okay, fine. Thank you. Thank you. First of all, I wish to thank Dr. Jose Putur and organizers for inviting me to share some of our views on heavy metal toxicity and heavy metal biology. I also would like to congratulate Dr. Jose Putur for organizing this meeting in Indian soils for the first time. Our team has been basically working on various aspects related with heavy metal stress biology since almost 1980. I initiated working on this way back in 1980 as a doctoral student. I was evaluating impact of cobalt and nickel on and also silver on flower physiology, especially on chrysanthemums. We were trying to evaluate impact of this on development and senescence of capitulum. We were using cobalt and nickel to curb the production of ethylene, while we were using silver to prevent the action of ethylene. Subsequently, after I joined as faculty in Delhi University, Jamia Millia, and involved in various facets of heavy metal biology. And today I'm going to share with you some of the aspects which we have been working on. Now, these are some of the scholars who have basically worked actively, and these are the people who educated me in a number of ways on various facets of heavy metal biology. Today, I am going to focus more on the work which is basically carried out by Shemia Emil and if time permits Shabnam. Now, I basically initiated as a faculty with Alia and Sandeep Arora as the initial people who worked on impact of heavy metal biology, and we've done very good. Those people have done a lot of work. In fact, our team has shown for the first time that proline gets produced under heavy metal stress, and we are able to find out the mode of production. And in fact, Sandeep has demonstrated how light promotes production of proline, and we were able to evaluate the impact of heavy metals on these aspects related with photosynthesis, ROS, and so on and so forth. Now, I'm sure most of you must have already Hello. It's
Sir, am I audible to you?
Yeah, you are audible. Yeah.
Sir, please share your screen. You just got disconnected. That's it.
Okay, fine. Is it all right now?
Yes, sir.
Okay. Now we have. Now it's clear?
Yes, sir, it is.
Okay. Now, here I was trying to focus on how exactly the heavy metals are present. They are basically present ubiquitously in the biosphere. And we know how exactly they are influencing biophysical and biochemical aspects related with living beings. And we are basically concerned about heavy metal toxicity now more at the level of biosphere and ecosystem dynamics, because now UN is basically concerned about restructuring the ecosystem, in fact, rebuilding the lost ecosystems. And at the same time, we're equally concerned about food safety and safe drinking water. So therefore, heavy metals obviously is playing a very important role. It's basically important for us to take care of these heavy metals, try to curb the production of these heavy metals. Why is the screen not? Okay. Although we know that heavy metals have negative impact on the health of humans and ecosystem, we cannot live without them. We know we are having essential heavy metals which are playing very important role in our physiology and biochemistry. At the same time, although there are several heavy metals which have nothing to do with, to the best of my knowledge, things, because I don't think there's anyone in this August gathering who doesn't use a mobile, because we live with them. Now, there are things which are there around us always. So we have been trying to understand how exactly plants regulate the fate of heavy metals. Nature has given various modes to curb the concentration of heavy metals in soils and also in water. Now, we have been trying to look at various things that are related with exogenous as well as endogenous means adapted or evolved by plants to curb these activities. We know plants do release more than 30 to 50% of its photosynthate in the form of various range of organic molecules. Now, some of these organic molecules which are released into the rhizosphere, they might be helping in chelating various kinds of heavy metals. Now, some of them may be able to reduce these heavy metals into some form which are less toxic, or they could be more, they're inert. No, and most importantly, we know that these organic molecules released by the plants, they basically try to take care of the microbes which are present in the rhizosphere, and they in fact play an important role in the dynamics of biosphere, which is there in rather I'll say below ground biodiversity. And we know these microbes in turn play a very important role in biotransformation of heavy metals. At the same time, they also play an important role in chelation of these heavy metals. Now, in addition to this, obviously, when you come down to the root surface, it's the cell wall which could be playing very important role. There are various components of cell wall which do have active groups which have capacity to sequester heavy metals. And then we do have plasma membrane bond dehydrogenases, which could be playing very important role in generation of inert materials from ions of heavy metals. And today I'm going to focus my attention more onto this aspect. Now, when you're looking at endogenous, we know endogenously we are finding organic molecules which might be chelating these heavy metals. There's a possibility that these heavy metals may be reduced to inert state. We are having phytochelatins and metallothioneins sitting over there. We do have the dehydrogenases which could be helping in converting these into inactive state. Maybe I'll try to throw some light on this if time permits. And we're able to also demonstrate very specifically that photosynthetic electron plays a critical role in curbing the toxicity of heavy metals by reducing this particular thing to a significant extent. So my focus would now be basically on how root system generate nanoparticles. Now, our group was approached by some of our collaborators to work on impact of silver and gold nanoparticles on biochemical and biophysical aspects of plants. So while doing that, we use salts of silver and gold as controls. To our surprise, we discovered that the root system of plants have potential to transform the color of these particular things. For example, here I'm basically projecting out the plants whose root system is placed in solutions of gold. As basically, as you can see, we are basically having 0.5 and 1 and 2 micromoles per ml of auric chloride, which is basically which is there in these particular solutions. Now, within few hours, you will be able to see conversion of these particular the colorless, maybe pale yellow solutions into rather they are clear solutions. They are basically being transferred into converted or biotransformed into some kind of colloidal and colored materials, colored solutions. And generally, we do find purplish color. You can find various gradations of purple color, except in Vernonia scenario. As you can see there, in this particular case, we're even able to find the goldenish colloidal material, which you can find over here. Now, we know that the color change, whenever the gold solution turns purple, it becomes reddish. We believe that it could be because of generation of nanoparticles. In order to check whether it is actually the root system is in a position to really generate nanoparticles, the first thing which we have done is to look for absorption spectra. We plotted absorption spectra and we have realized that we are having absorption peaks, let me say plasmon resonance bands, which are specific to gold nanoparticles. Now, which basically come in the range of about 500 to 600 nanometers. Generally, we might get peaks somewhere around 540 to 550 nanometers in most of the cases. The number of plants which are there, and of course, once we have realized that it is basically that we are having specific peak, it gave an indication that probably we are having nanoparticle synthesis. So therefore, we immediately went to their characterization. We have gone through, we have tested these particular things under transmission electron microscope. Now, as you can see, we are able to find distinct nanoparticles in these solutions. Whatever colloidal matter which is basically being formed, these are having distinct nanoparticles. These are the 4 plant systems from where we have basically taken these things. Now, when we are looking at the side pattern, that is selected area electron diffraction factor, we are able to find distinct rings, and these particular rings basically correspond to Bragg reflections for face-centered cubic metallic gold. And this in fact implies that these nanoparticles are crystalline in nature. We have also looked at the energies, the EDAX. We have evaluated energy dispersive X-ray, and then through these particular things we could detect the ions which are associated with this, the metal which is associated with, as it is clear. We are having distinct peaks of AU in all these cases. And of course, the peaks of carbon and copper, they have basically arisen just because we have used carbon-coated copper grids for loading the nanoparticles. These are the things with, we have got similar results with almost all plant systems which we have tried. So therefore, we know that the plant, the root system of plant is able to biotransform gold ions, the other auric ions, into auric nanoparticles. So we looked at the PXRD analysis. Now the PXRD analysis further given the clear indication that we are having Bragg reflections, which are basically which are specific. Whatever things which I'm showing over here, these peaks are specific to the gold metal. In fact, AU0 state of gold, and of course we are basically having that FCC metallic AU over there. Now, these things are basically corresponding with Joint Commission of Powder Diffraction Studies. So therefore, we have confirmed that these particular things are nothing but gold nanoparticles. So then we have evaluated the impact of this particular thing on silver. We basically then use silver salt. We have tested that the root system has potential to basically convert silver ions into silver nanoparticles. We have used silver salts of silver nitrate here, as is indicated. We have used 1 and 2 in the first, and of course we have also used 0.5, and these are basically so many micromoles per ml we tested. And as can be seen, we are able to see conversion of clear colorless solutions into brown colored solutions wherever you are having silver over there. Again, the degree of change in color depends on the strength which is available with them. Many times this is also depends upon the developmental stage of the plant system which we have used. Now again, as we have talked about in the earlier case, we have looked at absorption spectra, and in this particular case, we are able to find plasma resonance peaks somewhere in the range of 400 to 500. In the range of, in case of gold, we are having between 500 to 600, generally around 550 or so. Whereas in case of silver, we are getting somewhere around 420 to 430 nanometers. We could have some degree of variation. That's because of several factors that are basically coming up. Maybe if time permits, I'll discuss about that as well. Now, again, we have looked at these nanoparticles. We have evaluated them under transmission electron microscope. We can see beautiful, distinct nanoparticles, which are in the size range right from about 2 nanometers, it is going up to 100 nanometers, maybe generally about 50 to 60 nanometers. Sometimes we may go up to 100 nanometers. Again, here we are having the side pattern clearly shows distinct bands. But in case of gold, we are able to find only one speciation state. We could get only AU0 metallic state. Whereas in this particular case, we are able to find 2 states. We are able to get Ag0 state and Ag2O state. So that means you are having a portion of silver in the form of Ag0, that means in a metallic state, whereas in some other cases, in most of the things may be converted into, it is getting oxidized. Therefore, you are able to find Ag2O nanoparticles. And of course, the EDAX clearly indicated that we have silver in these nanoparticles. And of course, we have carried out with different plants. We got similar kind of results. And similarly, when we're looking at PXRD pattern, this whatever peaks we're having, these peaks corresponded with the face-centered cubic structure of AG0 as well as cubic geometry of AG2O. So that means oxidized state as well as AG0. Both the things we are able to find over here. Therefore, the number of peaks which you are finding in this particular thing is much larger than what you are seeing in case of gold. Our next step is obviously we have whatever I so far talked about. I talked about something, the metals which are precious. Now, and generally we don't believe that these metals, gold and silver, have any role to play in biological systems. Maybe they're having a negative role, and in some cases it may have other roles. But as far as iron is concerned, we basically wanted to see what is happening to the other heavy metals. The first thing which has come to our attention is basically iron. So therefore, we used perchloride. We tested if root systems have potential to convert this, and we are able to see the root system is able to convert instantaneously, kind of within few hours. It is able to transform this particular thing into colloidal matter, and this colloidal orange complex, it starts settling down to the base. Initially, you can see on the surface of the root. Subsequently, they start settling down. So, unlike in case of gold and silver, in this particular case, we don't get homogeneous solution. Here, you are basically having the complexes are basically being formed, and they are getting sedimented down. And you can, however, see this particular thing on the surface of root system very clearly. We looked at the transmission electron microscopic studies, and we are able to find nanoparticles. But however, the nanoparticles which we are finding over here are amorphous in nature. We don't have the distinct nanoparticles with clear structure as you are finding in gold and silver. In this particular case, we are having amorphous nature. We do find nanoparticles. We will be finding various shapes, and it's more amorphous, whatever. Unfortunately, in some cases, we were lucky, we were fortunate to get some micrographs which are beautiful. Only thing is, we need to have patience to look for these particular things. We are able to have some things wherein we are able to find good nanoparticles. Of course, these things stay for very short duration. Subsequently, they have tendency to coagulate. They have tendency to agglomerate. You can see clear agglomeration of nanoparticles in this particular case. So that means the nanoparticles which are basically being generated are getting converted instantaneously into complexes. And these complexes in turn are basically, many times these complexes are nothing but the so-called plaques which people have been talking of from last few decades. Initially, when we have gone for these studies, we were not very clear about this. We were not aware of the plaques. But subsequently, we realized that those plaques are nothing but they are the things which are basically composed of nanocomplexes which have been formed on the surface of the root. And we have gone for scanning electron micrographs. We can on the root surface, we are able to find distinct nanoparticles. We have controls, and you can just see distinct peak of iron. We were fortunate again to get beautiful structures. We got micrographs wherein you can see the nanoparticles which are again agglomerating. And I tried to put I put the TEM picture along with this just to show that there is a similarity in both TEM as well as SEM pictures are clearly indicating that these are having tendency to agglomerate and form large complexes. When we've gone for PXRD analysis, we can just see it's the amorphous nature. We just don't get any clear picture. It indicates it. And of course,
please
then we
can
have
you
basically gone
summarize your
for
presentation?
Okay, fine. The PXRD analysis as well as TG, it clearly indicated that whatever iron complexes which we are having, they are basically made up of iron oxyhydroxides. And we initially thought that the microbes which are associated with the root surface could be playing an important role in this. So in order to rule out that possibility, we have carried out these investigations under aseptic conditions, and we got similar kind of results. And in fact, we have basically compared the production of nanocomplexes or nanoparticles by plants with having better potential to perform this particular thing when compared to other things. And of course, even under these conditions, we are able to find similar kind of structures without any problem. And in order to demonstrate that, of course, root surface is basically having something which is unique. It is having some kind of dehydrogenases. So in order to show that, in order to see whether the root surface has potential to convert triphenyl tetrazolium into formazole. And we are able to see that root system has potential to do this. Anyhow, I don't want to take up too much this particular thing. Of course, we did get similar kind of results with several other things. This is with titanium and silicon. We were, and of course, you can see titanium, copper nanoparticles, then silicon nanoparticles, and also selenium nanoparticles. So we basically wanted to see, know what's the way it is basically doing. We then felt that the root surface is basically having enough reducing strength. In order to demonstrate that root is having strong reducing power, we basically used two clear-cut markers, indicators. One is ferricyanide, the second one is DCPIP. Now, the root surface was in a position to convert ferricyanide into ferrocyanate, and similarly, colored DCPIP into colorless DCPIP H2, reduced state. Almost all plants perform this job. And finally, we believe that yes, it is a dehydrogenase, plasma membrane bound dehydrogenases, which could be playing an important role in conversion of these particular things into the ions into atoms, and these atoms in turn get agglomerated to form various forms of nanocomplexes. Now, in summary, our findings demonstrate that all plant species across diverse taxonomic groups possess inbuilt potential to fabricate and release nanoparticles into their surroundings. We believe that the generation of metal nanoparticles by plants at root surface and the release into surroundings is a mechanism to exclude or avoid toxic levels of ionic species in state to metals. Okay, this seems to be excellent mechanism of phytostabilization. You want plants to counter toxic levels of heavy metals. And finally, I would say that our findings pave a novel, simple and economically feasible and eco-friendly scheme in case if we are standardized the kind of plant system, the developmental stage which we would like to use, we will be able to take these particular things forward to a significant extent. I think I will end now because I just wanted, in case if there's any doubts, I can come back to these particular things later on. But at this stage, I would like to say thank you for your kind attention.
Thank you.
I would like to invite questions.
Do we have a quick question from the audience? Okay, I have a quick question about the gold versus the silver. Did you see any more toxicity with the silver nanoparticles? The silver is involved in ethylene signaling in the plant. Any toxicity there?
Certainly. Silver nanoparticles are definitely toxic. Relatively, gold nanoparticles are more or less safe. This, of course, we have carried several investigations and also published, wherein we have shown that silver nanoparticles are photosynthetic events.
Okay, thank you very much. I think in the interest of time, we have to press on, but thank you very much for your presentation.
You're welcome.
Excellent. Our next speaker is Ebin Sebastian. The title of their presentation is Eco-Friendly Synthesis of Phenolics-Coded Magnetic Nanoparticles for the Reduction of Cadmium Accumulation in Rice. Yeah. Awesome, thank you.
Yeah.
Liz, just a minute, a small announcement. Some participants were asking for anything to be bought, to be taken home. So in the registration counter, we have kept certain spices and cashew and all. The price is displayed. If you can order those things by coffee time, that will be available to you by 4 o'clock if you want to buy something. The vendor has displayed over there. the items. If you like those, you can order directly in the registration counter. Thank you.
One more slide is there.
Okay, while I've been setting up, students, think of some good questions, yeah? There's lots of stuff here. Rice, cadmium, growth, toxicity. Come on, think of some questions, please.
Good morning and welcome to my presentation. Today I will be discussing about synthesis of a magnetite nanoparticles in a green way. But magnetite nanoparticle is not only good for reducing cadmium, it is used in a wide variety of applications. So let us start with So cadmium contamination of rice is a significant environmental and health concern. This research explores the use of phenolic coated magnetite, a novel material, to effectively reduce cadmium uptake and accumulation in rice plants. So this talk mainly covers some general aspect as well as some of my research which introduces use of magnetite for reduction of cadmium uptake in rice. So there are some cadmium pollution sources, mainly industrial activities, mining, agricultural practices, and all these contribute to cadmium accumulation in rice. In India, there are 4 places, mainly in Orissa, then Uttar Pradesh, Haryana. These places are mainly facing challenges, but there is less rice paddies. And main challenges in Kerala is because of the application of fertilizer. And we are neglecting it, but our soil laboratory researchers are pointing out that the fertilizer containing more than permissible level of cadmium. So there are some health risks with the cadmium. Consuming cadmium contaminated rice can cause some serious health issues like kidney damage and increased risk of cancer. So many countries have the strict regulation of cadmium for 0.2 milligram per kilogram is the limit. So if cadmium exceeds beyond this limit, it is creating a health issue. So these are main challenges with cadmium: kidney damage, bone weakening, and cancer risk. So there are methods based on soil and phytoremediation and breeding strategies. All these were already discussed in a review article. So we can use manipulate cadmium transporters, and we have a transgenic approaches. We have bio and phytoremediation approaches, cadmium complexation approaches, soil amendments, then fertilizer selection, also soil dressing and bio-augmentation. These methods are successfully applied in the various fields. So eco-friendly synthesis is nothing but it is green synthesis, and magnetite is the important thing is here. Magnetite, it is a naturally occurring iron oxide and it have many applications. Developing eco-friendly synthesis method is crucial to reduce environmental impact, and it enables sustainable production of these versatile materials. So the magnetite have magnetic properties. It is a ferrimagnetic material with unique magnetic properties, making it useful for data storage and magnetic sensors. So catalytic activity, it have the chemical structure of magnetite, allows it to act as a catalyst also. So we can use it for various application in remediation of chemical pollutants also, like pesticides, dyes, all those. So in biomedical, it is used as a promised target for drug delivery. And it is also used in magnetic resonance imaging, hyperthermia, cancer treatment. So the molecule is very important molecule. So conventional method include high temperature route is there, wet chemical techniques are there. So in both processes, high energy requirement and toxic precursors, these are the main problems. So conventional method lack sustainability and environmental friendliness. So when we look into eco-friendly synthesis techniques such as green synthesis, we have an environmental impact. So reducing carbon footprint and minimizing waste produced during magnetite synthesis crucial for sustainable development. It also helps to resource efficiency. It aims to utilize renewable and non-toxic precursors and streamline production processes. It is also cost effective and it has societal benefits. So developing green chemistry solution for magnetite products promote healthier environment and a better quality of life. So in the green chemistry, there are certain principles: atom economy, renewable feedstocks, and benign solvents. Atom economy is, we should clearly think of it, because what are the solvents we are using and what are the breakdown product we are getting during a synthesis process, synthesis as well as during the application phase. So we must always be synthesize. Even if we do a green synthesis, there is a lot of problems are there. Some of the reactant will be still in the media, so we should be aware while we go for a green synthesis. Also, when we carrying out a degradation of some chemical contaminants, we may be just biotransforming it or just transforming from one state to another state. So the plant may grow and there will not be any toxic effect. But what are the byproduct it formed, that it may also have some health effect or environmental, we should think about that one also. That is atom economy, what we use and what we produce, and there should be a balance. Then there should be renewable feedstock and benign solvent. And in eco-friendly synthesis, we always use water or ethanolic reagents, so there will not be much trouble. Mainly we focus on water. So the proposed eco-friendly synthesis is bio-inspired and leveraging natural sustainable precursor derived from plant extract. And we use aqueous medium and utilizing water as the primary solvent, avoiding hazardous organic solvents. We use also zero energy in fact, employing mild reaction condition to reduce consumption and emissions. Also, we targeted on waste minimization, designing process to minimize byproduct and enable efficient recycling. Reuse. So when we now we will look on to synthesis and characterization of phenolic coated magnetite. It should have a magnetite core. So magnetite nanoparticle provide magnetic core that can be easily separated from the environment. This is the one of the most important aspect. So when we use for environmental remediation or removal of toxic pollutants from the water media, we can easily separate it using a magnet. Then phenolic coating, it improves the surface area. So it improves our adsorption capacity and selectivity of the material towards cadmium ions. Then we generally characterize it with some techniques like XRD, FTIR, SEM. We basically use these techniques in our study. So when we generally speaking about a magnetite, it should have a particle size of 10 to 200 nanometer size, and it should have a cubic structure, and it should have a strong ferrimagnetic behavior and excellent colloidal stability in aqueous media. It should be there. So I used hevia as a source of phytochemicals for this synthesis because it is I come from middle Kerala, there rubber plantations are there. So also it contribute. It is not a simple thing that it is a billion-dollar business there. And also around gold, China, Thailand, Malaysia, these are the Indonesia, these are the major countries. But lot of rubber bark is going as a waste, while during its processing for a wood furniture, etc., this bark is removed. And also during regular tapping also, it is coming as a waste product, and nobody considers. So when I took a water extract of this plant, I found it is almost similar to a tea. So means it is brownish, full rich of phenolics. And if we do some biochemical analysis, all those, another trouble is there. We cannot simply evaporate it from water media because of the high molecular weight. So there are some challenges with that hevia, but it is a good material of interest to explore for various environmental sustainable activities. So I just took water extract, and one of the things we should notice is first one is the phenolic extract B, and C, it is a colloidal solution after green synthesis. So it is better always it is in a colloidal state to increase the activity of the particle. Then we checked with a simple magnet whether it will be attracted to a magnet or not. It was attracting the magnet. So I just partially confirmed it is a magnetite nanoparticles. So while looking into the same picture, we can see various sizes of the particle because of already told by Partha Saradi, agglomerization. But our target agglomerization is okay, also good. We can use it for environmental application. Agglomerization is mainly due to presence of phenolics as well as some mainly phenolics and a protein, because when you do a crude extract, protein will be less because it is degraded in the bark, but mainly phenolics it will help in agglomerization. Then we went for EDS analysis. We found that there is iron is there. Then after that we went for what are the chemical responsible for surface coating. We identified many benzoic acid derivative, means phenolic derivatives. It identified through GCMS. We just take a methanolic extract, say around 6 hours we kept in for stirring, and we identified this many peaks are there. So these were the chemicals responsible for agglomerization as well as mainly agglomerization coating, particle for the coating. So we went for adsorption of cadmium, how it will interact with the cadmium. So phenolic groups on coating provide chelating sites that bind strongly to cadmium ions. So also magnetic separation, the magnetic core allows cadmium loaded particle to be easily removed from the environment. Then regeneration, we can also regenerate because we can use HCl or some media, then we can get it removed from the magnetite and allowing the material to be reused for multiple cycles. When we look into XRD picture, it was not showing a crystalline nature, but there are peaks of magnetite also. So we after this picture we got it is in a semi-crystalline nature, not in crystalline, but it is in a semi-crystalline format. There was another one more interesting thing we got when we went for vibrating sample magnetometric analysis. We found that the hysteresis loop is almost a single line, means from ferromagnetic state it went to somewhere else, means it the phenolic coating may be giving it a bigger size and agglomerization, but it behaves unlike a normal magnetite. It was showing a behavior of a super paramagnetic nature. Super paramagnetic means they are nanoparticles, but they lose magnetic activity when we remove the media. That is why this hysteresis curve is in this fashion. So when next we went for adsorption studies, and we conducted two types of adsorption, Langmuir and Freundlich, and there was a strong correlation, means we got R square value near Hevia magnet. We also compared it with a normal magnetite also. In the case of normal magnetite particle, which was commercially purchased from Sigma, it was 0.78 only in case of Langmuir isotherm, but our particle had shown both in Langmuir and Freundlich. So we concluded that the particle is having a multi-layer adsorption, not in a single layer. Single layer means just binding to the surface, and in this case it is coming in a multi-layer fashion. So after that we went for greenhouse studies, and we checked for cadmium uptake and reduction in rice. The phenolic cotton magnetite had no adverse effect on the growth and development of rice plant. We also noticed there is a reduction in cadmium accumulation, and we also during this we did a sand culture. So we used a magnet to take it out from the sand also. These are some of the results. So it had a growth stimulatory effect in the soil. Also, when cadmium retarded, the treatment with the nanoparticle had a significantly increased the growth under cadmium stress. Also, when we compare the nanoparticle with the cadmium and the nanoparticle treatment, we can understand that it is the effect of nanoparticle, because nanoparticle alone effect is still persisting in presence of cadmium also. We went for some basic biochemical parameters, malonyl dialdehyde, peroxidase, proline. All these case we had a positive result. And when we went to metal accumulation studies with cadmium, it had almost half amount of cadmium only reached to the rice seedlings. This much part is good with this kind of studies, but when we went for a long-term trial, we found that the yield is less. But plant growth is stimulated, maybe shifting to more biomass productivity. But when we grow for rice farming, all those our target is also on yield. But yield is less, and also we noticed some browning of the tissues. It is then when we plucked the root, we found out that there is iron plaque formation in the root. So That was one of the drawbacks of application of magnetite into the soil. So it form iron plug and oxidative stress in the future, means when it goes to a higher level. And another thing is the downward migration of the particle. The particle not getting degraded, but it will, after repeated watering, all those, it used to come to migrate to the bottom of the container. So when we apply this kind of particle, we should think for ecological degradation also, because if we apply this kind of particle into the field, either we may contributing to the groundwater pollution. So I suggest this kind of particle. Magnetite is very good for removing cadmium from effluents, industrial effluents, all those in aqueous media. So we can easily separate it using a magnet. That is the best part. So basically, what are the outputs? Effective cadmium removal we have got. Phenolic cotton magnetite demonstrated excellent performance in reducing cadmium accumulation in rice plants under greenhouse condition. It also brings environmental sustainability. The material's magnetic separation capabilities and the potential for regeneration make it an environmentally friendly remediation solution. Future research will focus on optimizing synthetic procedure and evaluating the technology in the field trials for real-world applications.
All right, thank you very much. So we can accept questions now. First, students, if that's okay. First, yep, we have one there. Go ahead.
Thank you. It's possible using this magnetite in making an experimental new material like matrix, similar to activate carbon. Is making these like filter systems or something like that, because I don't remember if you said in this magnetite you only spray it in the soil.
Not spray, amendment. It is using as an amendment. Yes.
So it's like you put it directly in the root system.
With the soil.
In the soil, directly in the soil.
Yes.
But maybe you can build a new material only made by magnetite. Is possible?
Okay. We did not did that one. The field should be very high because it is mixed with the soil now. That is a good aspect of future research.
But maybe it's cheaper, this treatment.
Yes. But another case is solubility. In this case, it acts as a slow ion releasing agent. Magnetite will get mineralized. In the presence of these phenols, microbial activity might be increasing. That is why it is when we looked into the grain, iron content was very high. It got increased, and there was no cadmium under the field trials what we did. But overall yield is less. That is a problem because of iron plug.
Okay, well, thank you so much.
Thank you, Martha.
Thank you for the presentation. I have a question regarding the concentrations that you apply to the rice of both nanoparticles and cadmium.
Okay. Yeah, the nanoparticle rate was 0.1 milligram in that soil small study. In a higher study, it was 0.75 gram. And the soil quantity is, in case it was around 250 gram for small scale study, and for higher scale study it was 4 kilogram.
And for cadmium?
Cadmium was in micromolar concentration. We applied 3 times 250 micromolar cadmium chloride solution.
Okay, thank you.
We have one more question.
Hello, thank you for the nice talk. I would like to ask you some information about the potential creating action on other cations, bivalent cations, especially zinc, because we know that cadmium and zinc are often together in the soil or in the wastewater, or other bivalent cations.
Yes. This is also a future prospect only. I did not consider that matter. There is higher the chance it will bind with zinc also. Yes.
Okay, thank you.
Tea and chit chat later with this. There's clearly lots of good science to continue discussions, but we need to press on and try and keep to time. So do we have Raja? Ready? Hey, round of applause. Come on. So Raja's talk today is nano-enabled RNA interference and ultra-sensitive detection for comprehensive management of plant viral diseases. Okay. He's just taking the stage now. Everybody ready? Take it away, Raj.
Here. Okay, so here $220 billion loss year worldwide. So I was just checking today, like Elon Musk net worth was like around $270 billion. Last 2 days he got like a $27 billion gain. So with that money, I could buy like at least one part of Kerala, so like because it's a beautiful state. So here we go. $220 billion loss every year worldwide, so which means like plant diseases alone cause this much of effect in worldwide. So there's an analogy, India's half of total expenditure for the financial year is close to the $220 billion. So the plant diseases alone cause 12 to 16% of global food loss annually. So the WHO and then scientists and then other policymakers that concluded that this plant disease is going to be a persistent threat to the global food security. So when we are talking about the plant diseases, which is like most likely to cause by bacteria, fungi, and then nematodes, and then one of my favorite is viruses. It's not an organism, it's a thing, it's opportunistic parasites. So here's my topic, nano-enabled RNA interference and the ultrasensitive detection for comprehensive management of plant viral diseases. So my name is Raja Muthramalingam. I'm a postdoctoral scientist from Calicut Agriculture Experiment Station. So my big boss is here, Jason White, and then my another boss is Washington Da Silva. So he's on the third time honeymoon vacation with his new girlfriend. So sorry about that, he's a Brazilian. So yeah, this work has been like, we are doing this for several years with a lot of persistence and then perseverance, and then we finally succeeded to control the plant viral diseases, especially potato viral diseases. So here you can see the little bit evidence of the plant virus caused to the plants. So there has been like 1,300 plant viruses reported so far, which most of them, in fact, that every crab you grow for a food consumption, it means like the crab for non-natural agriculture practices, which is susceptible for, in fact, one or multiple viruses, which cause huge economic losses and then yield losses every year, worldwide. So people are listed like top 10 plant viruses in plant pathology. So one of them, like right now we are working on potato virus Y, which is ranked top 5. So the plant virus has simple architecture, just like a capsid protein, and then a nucleic acid, either can be a DNA or RNA, single stranded or double stranded. The only difference between plant viruses and animal viruses, it is the animal viruses has encapsulated, extra lipid layer with a spike protein. So which is, spike protein has receptor mediator entering system of the cell. Plant viruses doesn't have any receptor mediator. So its transmission by insect vector directly inject to the sap through the phloem cells. That's how the plant viruses get infected. So these are the common plant virus structures. So the spherical one is the predominant, and then also you can see like a rod shape and then filamented shape. The bacillus is very rare. So the another interesting structure you can see in the bottom. So the particle called Gemini, it has 2 spherical structures attached. So coming to the main business, so we are right now doing prevention and cure. So these are 2 main strategies that we are working on, we are focusing on plant viruses, especially for potato viruses. So prevention obviously means diagnostic. Diagnostics means like you can timely detect, and then at the same time you can control the spread of the viruses, which is significant in any of viruses. So we already experienced with the COVID-19 how the diagnosis is very important. And then cure means always like a treatment. You can do like a vaccination or medication, which is like a pre-treatment or post-treatment. So these two strategies we were executed, and then we succeeded already. So I'm going to show some success story here. So the problem with the plant viruses is there is no treatment no methodology for control plant viruses in the market. There is no kind of like a fungicides or bactericides you can apply and then control the bacteria, fungus, but there is no viricide in the market to control the plant viruses. And then how the people are controlling, they are doing like, viruses are basically spread through the insect vectors. So the people are using the pesticide to control the insect vector, which is transmit the viruses from infected to healthy plants. But this is not a sustainable strategy. So there is another mechanism or strategy that nature already give to us. So we have like a solution from nature in every time. So the nature teaches, so the mechanism is called RNAi mechanism, which is evolutionary conservative mechanisms mostly present in all the eukaryotic cells. So we will use the same RNAi mechanism to leverage with assistance of nanotechnology. So the RNAi mechanism is simply, as you can see that my left side. So this mechanism is generally how it works, I can explain. So when the genetic material from viruses enter into the plant, it activates the RNAi cascade of mechanism start from dicer, and then there is a lot of enzymes involving in that RNA mechanism. So at the end, it produces anti-interference, which is targetedly bind to the viral mRNA, which is suppress the virus replication, which is like completely eliminate the viruses particles. So here the important thing is, it is a natural process, but why then plant getting infection symptoms, even though it's presence of natural mechanism? The virus is a brilliant thing, opportunity parasite, even better than our human beings. So it can evade several antiviral mechanisms. So they have like several antiviral mechanisms. So simply then evade from the natural defensive mechanism. So we need to give boost, like something, so we can provide assistance to increase the plant antiviral mechanism against the viruses. So the double-stranded RNA, which I showed here, so we synthesize from the particular genomic region from the potato virus Y. We tried everything, almost every genomic sequences, particularly found only one part of genomic sequences works really well. So here we do like foliar application, just spray of double-stranded RNA in the particular genomic region, and then we saw controlling suppression of viral replication in the treated plants. So the problem here is, so generally we mechanically inoculate double-stranded RNA to the plants. So this is not going to happen with field trial or in a commercial level. So we need to find out some vehicle or carrier to deliver the double-stranded RNA to inside the cell to increase the antiviral mechanism. So that's how we need a nanocarrier to deliver the double-stranded RNA into the cytoplasm. The cytoplasm is where all the antiviral mechanism is happening. So these were the novel nanocarriers we synthesized at our station. Everything is new, not reported before. And then we synthesized like more than 10 nanomaterials, and then we found 2 of them really works good, means it delivered double-stranded RNA, which gives like 100% virus suppressions. So the fabrication, synthesis of nanomaterials is entirely different from normal synthesis process. Some material takes within 5 to 10 seconds to synthesize the material. Some material takes at least 2 days to synthesize the material. So it depends on how much cargo loading capacity. So we can alternate, we can engineer the nanomaterial nanocarrier to deliver the double-stranded RNA. So this is a normal process, how we do a nanocarrier application to deliver double-stranded RNA. So initially we infect, So for the post-treatment, this is generally we call as a vaccination effort. So we foliarly spray the nanocarrier with double-stranded RNA to the plant, and then we infect the viruses, potato virus Y, and then we take a measurement. Mostly we use the qPCR to quantify the virus concentration in the plant. So these are the results we obtained from the treated plants. So as you can see, the red boxes alone, so that's the one with particular concentration of nanomaterial along with the double-stranded RNA. So we observed it is the first time we observed 100% of suppression, virus suppression in the treated plants. So there is some reports, early reports shown like a 15 to 30% of virus suppressions. So we are lucky, I don't know, like lucky is not word. So we were able to achieve at least 99.9% of virus suppression with mostly it's a concentration dependent. So we thought it's a novel of very, very novel work, and then we file a patent. So it is now in a bending position. So coming to the diagnostics, so we partnered with 1215 Molecular Diagnostics. So Jason also is a convener of that company. So the product called Verilize. So why we need a diagnostic, even though we have like existing diagnostic methods like ELISA and then PCR methods. So as you know, like ELISA and PCR methods take longer time, and then it is very expensive. Because I personally experience it with the PCR machines, because in my previous postdoc, so I usually do like thousands of samples with the virus infection. So it takes like a month-long process, RNA isolation, cDNA conversion, and then you need to run the gel PCR. It takes like a month long. So we thought that the company has some new technology using nanomaterial, carbon nanotubes as a sensing with the probe of DNA. So we contacted, Jason invited them and give a demo to our station, and then we contacted him. So we almost like developed, we almost like developed the technology. So it's been like 1 year almost. So they are about to commercialize this product. So what the advantages here compared to the PCR machine and then ELISA is, this technology is very fast. It takes only 8 to 10 minutes to analyze the results, and then you will get to know whether they're positive or negative, and then also it gives a quantitative results. So this I can explain how this sensor works. So this is basically an electrical sensing based, which component the sensor will have like a carbon nanotubes, and then it will have the probe sequences, which the probe means that primer sequences which you use for the PCR amplification. The same sequences we use as a probe. When it targets point into that probe, so it gives very minimal electrical signals, which like manipulated as a LED signals, and then also they have like algorithm to generate a quantitative quantification methods. So these are the designs of their instrument. So as you can see, this is like a very small, tiny instrument. They have like a sample holder, and then it comes with a sensor. Once you put the sample with the proper buffer, they close with the lid, and then the whole lid and sensor will go into the devices. So within 8 to 10 minutes, it gives a LED signal, either you have a positive or negative in that samples. So they compared with qPCR already. So they achieved the specificity and then selectivity, sensitivity more relevant to the qPCR results. So these were the latest results. So they done like several experiment optimization along with the qPCR. They achieved same sensitivity. So right now they are about to patent this work. They are about to commercialize when the patent is done. Thank you so much. These are my beautiful people from our station. I thank you, USDA, for the funding support. If there is no funding, there is no research. If there is no research, there is no researchers. If there is no researchers, there is no civilization. I would like to thank L University for characterization facilities. Thank you.
Okay, I'm going to check the whole of the room. Any questions? Okay. Okay, Benoit.
Very nice talk and very interesting work. So for practical applications, is there any issues related with the stability of the RNA and just the embedding of this RNA into particles? Can that enhance somehow the stability of RNA?
Okay, so your question is about the stability of RNA when combined with a nanocarrier. Am I right?
Yeah.
Okay. So we tried several like metalloid nanomaterials. When we are combined with metal and metalloid nanomaterial, before we came into the biomaterial, we have seen the efficiency of RNA was completely controlled because of some interactions between metal and metalloids. So when we switch into biomaterial, so most of the material what I shown here were completely a biomaterial. So we didn't see any inhibition or we did like several experiments. I mean, the functionality of gene we tested with the PCR. So it shows the 100% stability when they combine with the biomaterial. You got it? Thank you.
Okay, right. Students, you've only got four talks left to ask a question. Time's ticking. Have we got one now? Anyone? You look a bit mature for a student, Jason, but take it away.
Well, since no students have questions, so a thought popped into my mind. Raja, as you know, we have a mosquito virus surveillance program in the state to test for Eastern equine encephalitis and Zika and West Nile. The process of going from a mosquito to actual virus detection is about 4.5 days. I'm wondering if, I mean, this is completely off the wall for this meeting, but we should think about whether this type of approach could be applied to an insect
that's
insect model.
Right, that's interesting opening. So I already discussed with those points with some of the entomology people.
Oh, okay. Well, you're on it then. I'll stop.
Yeah, thank you so much for the question. Yeah.
Okay, thank you very much for an excellent final talk, and I'd like to thank all the speakers for the session. We'll go for tea now. Thank you.
You can
unplug this.
Yeah, you may.
Oh,
There is another.
There is another. It's perfect.
Can I copy it, or do you want to use it on your pen drive itself?
Sorry.
Do you want to use it on your pen drive, or like PPT will work on this this one?
Oh, it's okay. PDF is okay. I don't have it.
Kindly note, all the presenters and the participants can download their certificates through the scanner code which is available in your ID card.
Oh, I'm in the breakout room.
I tried to send this file via link. It's 500 megabytes, and apparently you're having problems.
Sir, may I help you? Can you just repeat what you told right now?
Oh, this is Barry. I attempted to send the file via an upload link.
Yeah, we got it.
And I had
Yeah, we got it.
Okay.
Yeah.
Is it work? Okay. Do you have it loaded? I can go live, or you can use the recorded.
Can you present online, or is there any other option?
I can present, or is the recorded version? Do you have that, and does it work?
It's like the audio is coming, but no video.
Oh, okay. Let's do it live then. That works.
Yeah, it will work. You can go live.
Okay, thank you.
So we are having a 10-minute break time, so we will resume back, and I will let you know. Like, be here for some time. Like, it will be possible, right?
Yeah. Let me just share my screen real quick. See if you see it.
Yeah, I will tell you to share your screen.
Do you see it?
Yes, sir. It is.
Very good.
Yeah.
So I'll stop sharing and go back later.
Yes, sir.
Thank you.
So this is the time for tea break. If you guys want to have some refreshment, please go outside and have some snacks and tea. Thank you.
Thank you, sir. Thank you all. Thank you for accepting this unorthodox presentation. This is getting on the fringe of phytoremediation. I am a practitioner in the discipline. I'm a geologist with education in geology and biology with a focus in nature-based solutions. This is privately funded research from a petroleum company that we work with. We just completed an unpublished white paper, PFOS sensing by an inaural organism, Phytotechnology polycephalum. We just did this last month, so this is fresh off the press. Our objectives here were exploring the potential of this organism, Phytotechnology, as a potential biosensor for PFAS. And in this case, we're using PFOS as a model compound. That's perfluorooctane sulfonic acid. Just showing here the slime molds in the, I'm going to kill this laser pointer. Just to talk a little bit about the slime modes, where they fit in the phylogenetic tree. They're over here to the right, near the animals, fungi, and plants. So we are working with a primitive organism. So a little bit more about Physarum, and I'll call it Physarum from now on. These are called colloquially as slime molds. They're in the phylum Amoebozoa. There are different, depending on what literature you read, there's different classes, either Myxogastria or Myxomycetes. These organisms favor temperate, cool woodlands. We see them out in the field, but they're also widely cultivated for various lab studies. The feeding stage that you see off to the right is called the plasmodial phase. This is the phase that's expanding and looking for food. The sole purpose of this organism is essentially to search for food, while the dry dormant phase is called the sclerotium. For this study, we use both phases. This organism is a fascinating creature. It's well studied due to its proto-intelligence, its ability to navigate mazes, and to also to search for food, and also to react to different environmental stimuli. We see quite a few studies with chemotaxis and mechanosensation, and also studies that focus on cellular differentiation. Some precedent work. So back a few years ago, AECOM, as a proof of concept, developed a growth and feeding experiment. This is basically, we're trying to understand the organism and the limitations of cultivating it, and also studying the behavior in a controlled environment such as Petri dishes. This evolved into a proposal, a novel surfactant biosensor. At the time, we were studying how this organism reacts to various soap-like compounds and surfactants. So off to the right, this is a study, an image capture from a study by Murigan and his associates. And this is a time experiments, time lapse, showing various hours. And these are weighted discs that are added to a 2% agar solution. Physarum is in the center of each of these dishes. And you'll see that this organism is moving over to the heavier loading within the agar. So Murigan and his associates, they essentially built up a hypothesis, which they later substantiated, that this organism can sense surface tensions. So after reading this paper, I got to thinking, well, maybe this organism can sense surfactants, such as PFOS. So here we are. We use a very simple experimental laboratory design. This is a binary bait situation where we're using Cheerios. Cheerios are, of course, a breakfast cereal. We selected Cheerios over other cereal grains such as oats due to the uniform size, and they're also very easy to maneuver around, whereas oats have different sizes. So we were concerned about having different sized baits because the Physarum may react to that. We're dosing one Cheerio with a PFOS solution, either 10 nanograms per liter or 100 nanograms per liter, while the other Cheerio is dosed with plain water. All our work is done in a polystyrene Petri dish, and then we're also using wet, moistened filter paper. We experimented a bit with 2% agar. I'll talk a little bit about that as well. One of the key points is because this organism is light sensitive, we rotated the bait. So we used different bait geometries on these gridded plates. We had a total of 20 samples. So this is not a big statistical study, but we started with 20 total samples. Here's a summary of the test groups and the dose groups. 2 dose groups at 10 and 100 parts per trillion, roughly, PFOS. We started with 5 test groups. Due to economics, we moved down to 3 test groups. Then we also did time trials, and the time trials were done stop motion photography, where we used GoPro camera, and I'll talk a bit about that. Off to the right are some pictures of the baseline for the initial test groups or test trials. Just to talk about some of the equipment, we're using live and dried Physarum cultures. These are derived online through Carolina Biological Supply. These cultures are actually used by high school students. They're non-pathogenic. There's no harm in utilizing these organisms. We're using Petri dishes, Cheerios, sterile filter paper. In some instances, we are using an agar gel substrate. We have prepared the PFOS solution using a standard from a potassium PFOS salt. So we're working from the salt, we're dissolving to meet those specific concentrations. All the work was documented using iPhone. In this case, we're using iPhone 15 and a consumer grade GoPro Hero 10 camera that's shown off to the right. This camera got really hot. There's a very small battery, and for doing these time-lapse photographs, sometimes for 3 days, we found out the battery burned up real quick, so we used an ancillary battery, 12-volt battery source in one of these miniature power stations, and this all runs off direct current. So one of the things that we study is we wanted to classify the different Physarum movements. And this organism behaves, it will crawl all over the dish. So we wanted to quantify these basic movements. So a PFOS positive movement is shown in the first image to the left. And in this instance, the organism leaves a center position and it's on a filter paper, so it's a dried phase, it's hydrated, it moves to the PFOS. Very clear PFOS positive. Then sometimes Physarum does funny things. It can just grow and sit in a centralized location or move lateral. Within this broad group of movements, there's a movement called satellite movement, where it basically darts out, relocates, sits there, kind of centrally located to the off to the sides. It doesn't do anything. And then the other movements are ambiguous, where it tends to go all over the plate. Given time, it just goes everywhere. So the one document to any of these trials was the water positive movement. We don't have photographic evidence of that. So that would be where it actually goes to the control, Cheerio goes to the water. I've used a simple color coding to demonstrate the results. The red is a PFOS positive. So we're using the stoplight analogy that Steve Jobs used for Apple years ago. Then there's a amber centralized lateral growth. Water positive, we never saw, that would be green. And then there's this ambiguous movement I'm showing as lavender. So just looking at our hit rates, and I recognize this is a small population, the hit rates we're seeing a little bit better in the higher dose. So we're thinking this organism can differentiate between different dosages, between 10 and 100. In the future, we'd like to step it up and look at different ranges, maybe some other chemicals. And in the time trials, which were typically done on the gel. The time trials, we have basically 100%. These organisms move right to the dosed Cheerio. So I'm going to show here, this is a time-lapse trial. Now, early on, you'll see some condensation. These are covered Petri dishes. Off to the left, this circled area, now that's underneath the Petri dish. That is the PFOS dose Cheerio, whereas the Cheerio to the right is the water dose. The Physerum is on a filter paper, centrally located. We use calipers. We were very careful to orient this Physerum relative to the Cheerios, the same distances, because this organism tends to take the shortest path to food. It's quite energy efficient. So let's look at the video here. So these are 5-minute time-lapse photos, 5 minutes, then another photo, then another photo, and then you run it in stop motion. This is over roughly a 7-hour duration. So this is a pretty active organism. I'm going to show this one more time. There's time. It thinks there's a time where it's evaluating things. It's thinking about going to the other Cheerio. Then ultimately it goes to the dosed one. Next slide. There. We have literally hundreds of photos at different stages of growth. I'm showing some of the better photos at different dosages. So you do see that it's very clear how this organism moves. It leaves its central location, moves over, explores these Cheerios. And in some instances, it sits in the central image. It's sitting there on this Cheerio. Now it's digesting, it's eating bacteria that are on the Cheerio, but it's also sitting there where you want to study a little bit more. What is it? How is it interacting? Is there some type of, is it attracted to the carbon, or is it attracted to the fluorine ion? So what is it interested in? So these are ideas for future work. Okay, some challenges and lessons learned. I mentioned earlier that we played around with 2% agar and also live cultures. So you would take an existing Physerum culture, use a scalpel or an X-Acto knife, you cut that culture, you transfer it to new agar. And we ran into all sorts of contamination issues. The Cheerio to the north on top is a dose Cheerio with PFOS. Cheerio down at the bottom is just water, and then there's the Physerum culture in the middle. Those folks most of you will recognize this as penicillium. We actually did some sequencing targeting both the 16S rRNA bacteria gene, but also we did sequencing for bacteria. And interesting enough, this lacy rind that we see in the northern Cheerio, this is this crateriform colony. These are various bacteria that we find in the Pseudomonas group that are found in cereal grains. So there's no surprise. So when you're eating Cheerios, you're probably consuming this bacteria. Now, what's interesting, and I can't talk too much about this until I think we're going to talk about it at the Patel Bioremediation Symposium in Boston. We identified one group of bacteria that we find in fluoroacetate producing plants, which is really has us puzzled. And so we've got a fluoride tolerant bacteria that's showing up in our cultures. So we want to explore that a little bit more and get to understand that bacteria.
Barry, you hear the bell, I think, so that means you have 2 more minutes before questions.
Thank you, sir. So we see chemotaxis. We do see some dose sensitivity. We're starting to think a little bit more about using a neural organisms without nervous systems as biosensors for chemicals. There's been some work done in the past. There has, as far as we know, nothing's been done with PFAS. So we want to think about that a little bit more and craft up some future research. We're thinking about looking at co-solvency with PFAS and fuels. These are bridge compounds that connect polar and non-polar substances. We want to see how Physarum reacts. Also look at maybe different compounds, the short versus long chain. Is this organism reacting to fluoride ion? Maybe we look at that. One thing, AECOM, we're not a clean room environment. We're not a research company. We're an environmental engineering consulting company. So the idea of partnering with an academic that has the right environment for cultivating these organisms. And then the other thing we're thinking about is AI and or machine learning to better quantify growth. This is a whole nother world that we're all learning about. So there are novel ways to use computer-assisted platforms for gauging growth. That was the last slide. So the sponsoring team, Kyle Waldron is a big supporter of PhytoRem. He's been to a few conferences. We saw him in Chicagoland last year, and so he's always into novel stuff. Then Megan Kubash and Kim Brogan were our Physarum wranglers, and Dr. Barajas is always a keen scientist that provides interesting ideas. And I'm here for questions. Thank you all.
Thank you, Barry, for this interesting presentation. This is really fascinating work. All right, we have the time for a few questions. Students first. Are there any questions from students? Come on. Oh, okay.
Are you seeing the slime mold have any negative effects after it's interacting with the PFAS? Any like toxicological effects?
Now, that's a fantastic question. We don't. Now, depending on the temperature, we incubated right around 24C in a darkroom environment. And this organism does have a lifespan at those temperatures of about a week. So they go through a life phase, so they change. But I haven't witnessed anything that would suggest to me toxicity. But we aren't working in the parts per billion or parts per million range. So that brings up a great idea. Maybe the next time we start looking at higher concentrations.
All right, thank you. We have the time for one more question.
Hey Barry, Jason White here. Just a quick question.
Hi, Jason.
Interesting choice that you used a Cheerio. What do you think would happen if you used like an inert kind of carrier, like, I don't know, cotton or a piece of clay or something like that? Just remove the nutritional source altogether and just go with PFAS.
We've thought of that, and we might use a resin or some type of porous material, maybe a ceramic. But that's a great idea, and I think that's something we'll probably explore. Because like you said, you're minimizing the variables.
Yeah, very cool work. Thanks.
All right.
Thank you.
Thank you, Barry. And
thank you all.
All right. So let me call. We can go. All right. Thank you very much. All right. Our next presenter is Gregory Cohen. So Dr. Cohen is, sorry, I have to speak in the mic. So Dr. Cohen is professor in EPOC at the University of Bordeaux. So EPOC means in English Oceanic and Continental Environments and Paleo Environments. So Dr. Cohen is going to talk about dieldrin in pumpkins. So your turn.
Yeah, thank you very much, Benoit, for this kind introduction. I'm not professor yet, just professor assistant, but maybe soon I will get professor. So, yeah, today I'm going to talk about dieldrin accumulation and allocation pattern in cucurbita pepo, especially in zucchini. This work was a part of the Phytotechnology project, which was funded by the French Agency for the Environment, the ADEME, with Bordeaux Metropole and the county of Gironde. So, small description of the dieldrin, which is a member of the what we call the dirty dozen. So, dieldrin is organochloride pesticides called OCPs. It was used between the 50s and the 80s all around the world, mostly as an insecticide to control the insect-borne disease and the living of insects in agricultural soil. Unfortunately, this is a toxic and persistent compound, so it was classified as a POP by the Stockholm Convention, which permitted to ban internationally this kind of compounds in 2001. In France, it was banned from agriculture in 1972 for agricultural purpose, but even in 2024, we still observe it in arable soils. And unfortunately, there is an accumulation in plants from the Cucurbitaceae family, and we observe a concentration in the fruits which are above the maximum residue levels, the MRL, which has the maximum concentration which are allowed in the consumption products. So the question is, can phytomanagement minimize the transfer to the food chain of this kind of compound? Let's go a little bit backward on the historical context. In fact, in summer 2015, there was a discovery of the presence of OCP in zucchinis, namely dieldrin and chlordanes. This was discovered from the French Department of Food Regulation, which had the good idea to test some old pesticides in zucchinis. So it was a surprise to find such contaminants in food, even if they were banned from agricultural purpose use since more than 40 years. So at the end of 2015, there were complementary investigations on the soils to observe if there was soil contamination with these compounds, and of course they found these contaminants in soil. So there were few articles in the local press which was highlighting the surprise of everybody to still find this kind of compounds. And at the beginning of 2016, the states proposed an action plan, which was, as it's written here, to ask the market gardeners to grow adapted crops, even if they don't know which kind of crops could be adapted to this kind of contamination in regards to the OCP contents. Of course, the states enforced the survey plan to have a look on this kind of compounds, and especially on the market garden products. And they asked the market gardeners to practice systematic control of the production for the most accumulator crops, namely the cucurbits, when they couldn't move this production in other areas. Of course, you can imagine that on an economical point of view as well as a technical point of view, it wasn't suitable for the market gardeners. So, what is the situation today? After this discovery, the state has the good idea to have a look in the whole France territory, and you have some articles which were published last year from several journals, so I let them in French to not disturb the assembly. I'm kind of try to translate a little bit. It's saying that we are sitting on a sanitary bomb. They observe this kind of contamination even in organic farming lands. And some people were saying that the only alternative to this problem was to stop to spread this F-word compound on the lands. So there was even national review at the French television. So just to tell that it is an actual problem in the whole France and probably in the whole world. So Let's go back on the state of the art about the accumulation, the impregnation of dieldrin by crops. You have to know that the root pathway was for a long time neglected, since this is an hydrophobic molecule. It was considered that only aerial deposition could occur because as it is hydrophobic, there is no dissolution in the soil pore water, so the root uptake was neglected. However, several studies highlighted that the opposite with plants impregnation with organic contaminant could occur. Among the plants, the cucurbits show the highest ability to absorb, accumulate, and translocate the organic compounds from roots to shoots. We can observe different accumulation capacities, but there are several bibliographic gaps. Most of the studies were conducted with spiked soil. There were often no discrimination between the different organs. Most of the time it was just a distinction between the roots and the shoots. And lots of studies were conducted on plantlets. So the question remains, how dieldrin transfer from soil to plants for cucurbita pepo? What is the dieldrin accumulation and what is its allocation, in order to find out management solution and maybe remediation solution. So what we do, we start with the selection of the zucchini varieties, and we selected varieties with contrasted phenotype, especially on the fruit shapes, the color, and so on. So we selected 10 varieties of zucchinis, which are listed here. Anissa, Bellor, Mioza, Noir Maraîchère, Vert d'Italie, Aurelia, Gold Rush, and Floridor. Some of them were dark green, dark and clear green, yellow, and some of them were long, and one was round. Then the seeding was conducted in greenhouse. They were grown during 1 month. And then they were transplanted into pots on the real field condition with 1 plant per pot. We use an historical contaminated soil, about 100 micrograms per kilogram of dieldrin, and for each pot there was a sieving at 1 centimeters. We homogenized the soil in order to get as much as possible homogeneous concentration. We sampled every soil of each pot to get a precise ID of the concentration, and we buried the pots in the land in order to protect them from the heat and the pest. Concerning the harvest, we harvested the fruits individually when they reach commercial size, so namely 20-30 centimeter long. We harvested the plants after 3.5 months, and after that we separated the different organs, the leaves, the stems, the roots, the flowers, and the fruits. We washed all these organs with tap water, and we ground them to obtain a puree for the fruits and powder for the other organs. So we were working with a fresh weight for the fruits and dry weight for the other organs, which were dried at 30 Celsius degrees. Concerning the analysis, we conducted extraction with Dept during 48 hours. We recovered the supernatants after centrifugation. We had an internal standards for the analysis, and we conducted analysis with GCMS. So let's move on to the results. On the left side, you have the biomass of the zucchinis, of the 10 varieties of zucchinis, and as you can see on this graphic, most of the biomass is in the shoots. There is a small small quantity of mass of the roots, and depending on the variety and the production of the variety, we have a different proportion of fruits in regards to the other shoots organ. The graphic in the middle of the slide show you the different concentration in dieldrin that we measured in each of these organs. So at the bottom you have the roots in brown, just above you have the stems, then in dark green the leaves, in light brown the flowers, and in yellow the fruits. So as you can see, the dieldrin concentration are higher in stems than in roots. Much lower concentration were observed in leaves, and then fruits and flowers. So fortunately, the edible organ of zucchini's plants are less contaminated organ, which is pretty fortunate. Another interesting point is the variability of the dieldrin concentration in this organ. We observe that for vegetative organs, we have a variability of 2 to 3, while for the reproductive organs, so namely flower and fruits, the variability of dieldrin concentration is much higher, between 5 and 15. So that means that the dieldrin allocation in the reproductive organs is variety specific and not related to zucchini plant absorption capacity. We had a look of the influence of several factors on the zucchini's fruits contamination. So we can see variable and weak influence of these different factors. First of all, the soil contamination. We observed that the concentration of dieldrin in zucchini in the fruits were increasing at the beginning and then reached a threshold, and so a pretty stable concentration. And except for the Gold Rush variety, we had no significant relationship between this the dieldrin concentration in soil and the one in the fruits. The time of exposure doesn't seem to have great influence neither, except for 3 of them for which we observe significant tendency, for the Anessa, Bellor, and Noir Maraîcher, but as you can see also it's a pretty weak slope. And concerning the fruit biomass, there was no relationship between the fruit biomass and the dieldrin concentration, except for one, for the Floridor variety, but as I told you earlier, the Floridor variety was the only round zucchini, and as there is higher concentration in the peel than in the flesh of the zucchini, that's why when you have bigger fruits, you have less peel and more flesh, and so the concentration in dieldrin is decreasing. So that has a consequence on the phytoextraction capacity of these varieties of zucchini. As you can see on the graphic, the dieldrin is mainly stored more than 95% in the shoots, with around 40% in fruits, depending on the variety, around 35% in stems, and 25% in leaves. Concerning the extraction of the dieldrin from the pods, we measured that it was around 150 and 300 micrograms of dieldrin that was extracted from the pods, so which is between 4 and 10% of the initial concentration in each pot. So that means that we have 2 solutions which are offered to us and for the market gardeners. The first one is the selection of varieties with low accumulation in fruits. The advantage of this technique would be the ability for the market gardeners to sell their production, but the bad points would be that it would be a slow soil remediation. At the opposite, we could select varieties with high accumulation in fruits, and of course the production of the market gardeners should be destroyed, but on the other hand, we could have a faster soil remediation. We also try to characterize the allocation of dieldrin in the zucchinis and to try to observe the link between the different organs of cucurbita pepo and dieldrin accumulation. And what we observed is a high relationship between flower and fruits. We also observed a relationship between roots and stem, between stems and leaves, but the highest relationship was between another high relationship was between leaves and fruits. That could suggest that the dieldrin supply to fruits is made by leaves. How could we explain that? The works of the Japanese team highlight the key role of what we call the major latex-like proteins. There is a specificity of cucurbit's major latex-like protein which present an hydrophobic cavity which could allow the formation of a complex of MLP and POP in the roots, and this complex could transfer to the shoots via xylem sap. There were about 21 MLP genes which were identified in zucchinis, and they also observed the influence of pH on MLP capacity to bind to the pops. So to conclude, we saw that there were different dieldrin accumulation in reproductive and vegetative organs of cucurbita pepo. There is low influence of soil contamination on fruits contamination. Up to 10%, around 300 micrograms of dieldrin can be extracted from soil for experiments in pots by zucchini plants, and means of management with concentration in fruits below the MRR and contaminated soil remediation with phytoextraction were identified. So what are the perspectives now? The main point would be to characterize the mechanism which could explain this allocation, this different allocation depending on the variety. One of the main questions is also what is the real bioavailability of dieldrin concentration in soil? How will this soil bioavailability dieldrin content will evolve in short, middle or long-term condition? And one of the key questions is also what to do, how to manage the above ground plant residue with high dieldrin concentration, and so many other questions. Before to thank you, I would like to thank all the partners who participate to this project, especially Dr. Marie-Cecilia Folder, who did most of this work. For sure, Dr. Michel Mensch, who helped me a lot in this project, and Dr. Félix Collin also, and all the students who worked with us during these last 3 years, and of course the market gardeners who helped us a lot. Without them, we couldn't access to a real historical contaminated field, and so thanks to them, and I would like to thank you for your attention.
Thank you, Greg. Great presentation. So we have the time for a few questions, and as usual, I would like to call. Yeah, please go ahead. Oh, all right. Yeah. Okay.
Very nice presentation. I would like to ask that eldrin, dry eldrin, they are a very, very toxic xenobiotic compounds that comes under the xenobiotics, in fact. So if we are taking some of the edible crop for the management, how does we are going to manage? Point number one. And point number two, there are so many microorganisms which has already been identified to take in their metabolism this kind of xenobiotics, many pesticides and hydrocarbons, in fact. So are we also going to incorporate some microorganisms which might help a lot for the remediation, which is already existing over there, what you told around?
Thank you for this very interesting question. Yeah, we were thinking about introducing microorganism. Unfortunately, as long as we know, there is no specific microorganism who is allowed to degrade the dieldrin and these compounds fast enough to get remediation, but we still should conduct an experiment about that. But the key question beyond that is if you do degradation of this kind of compound, what kind of toxicity will have the degraded compounds, which is a really key question. If you degrade them to a more toxic substance, it wouldn't be interesting. So, but this is one of our future ideas for the next project also. Thank you for this question.
I think we have another question over there. Yeah.
Okay, thank you. And based on your results and your perspective, do you think is it correct to affirm that dieldrin move more in the xylem and less to the phloem of the plant? And if you think that there are some influences by the apoplastic or symplastic uptakes?
Thank you for this question. Really interesting question. Unfortunately, I don't have the answer to that. So maybe Michel could have an idea, but...
In fact, we don't know how the major latex protein are moving and allocated between leaf, stem, and fruit. One of the key, it is a molecular determinant for all these mechanisms for allocation. And it may depend on the development stage of the plant.
All right, thank you. Please. One quick question.
One quick question. I'm very excited about your major latex-like proteins. This could be really interesting and exciting. Have you been able to purify these proteins and establish the binding affinity? Is this something you could express into the root exudates to bind and keep them in the soil? Or, I don't know, they sound really cool.
Yeah, I totally agree with you. It's something pretty cool. Actually, no, we don't know how to analyze these proteins. That's only the Japanese team which has this competence. That's why we're trying to have a collaboration with them to try to explain the differences we observed between the different varieties of zucchinis and to try to find out why there are several with high accumulation capacity in fruits and one with low. Probably the answer are in the genomic or the omics in general, but that's where we are.
Yeah, we could chat later. I have some ideas.
Oh, great.
All right. Thank you, Greg. So Greg is going to introduce our next speaker.
Thank you, Benoit. So the next presentation will be an online presentation. It will be conducted with Chen Tu from the Chinese Academy of Science in China. He's gonna make a presentation on identification and characterization of a biodegradable plastic mulch film degrading bacteria strain PB81 in the soil. So Chen Tu, if you hear us, the floor is yours. It's a recording, so he doesn't hear us.
Hi everyone, I'm Dr. Chen Tu from Institute of Soil Science, Chinese Academy of Sciences. Unfortunately, due to my late visa application, I cannot come to India to join the IPCA team and to meet some of the old and new friends with you together. But thanks to the organizers, they give me the opportunity to share with you some of my recent research online. Today, I'm going to talk about the identification and characterization of a biodegradable plastic mulch film degrading bacteria, PBAT, in the soil. Today, I'm going to talk in the following 4 parts. First of all is the abundance and the source of micro and nanoplastics in arable soils, especially in China. Then we talk about the changes and trends of microplastics after they entering in arable soils. And then for those microplastics in the soil, we're trying to introduce microbial degradation of these microplastics and to identify their mechanisms. And I should give some future perspectives at last. First of all, let's look at some backgrounds. As we may know, plastics, especially microplastics, have been made recently for many purposes of use. So after their usage, they were abandoned in the environment, and they can be found in atmosphere, the terrestrial and the marine systems. And due to the transfer from the food web, they can be finally uptaken by animals and then by human beings. One of our recent study from our group shows that we can detect microplastics in the human thrombus tissues. So recently, many publications have been introduced that micro and nanoplastics have been found and detected in remote ocean surface and coastlines, in marine water columns, even in very deep sea, and the soils, especially the arable soils, and also human bodies cells. Soil is an important sink of environmental microplastics. From the two very famous reviews, we know that the abundance of microplastics in global soil samples varies from several to 500,000, and it changes along different regions of the world and also varies with different soil types or land use types. And the main polymer of the different plastics also changes, and their size also changes along different regions. According to a survey jointly initiated by our group and Professor He from East Normal University, we found that the microplastic abundance in 30 farmland soils of China varies from 25 to 2,068 items per kilogram, with a mean of 358 items per kilogram. The microplastic abundance varied also among different regions of China from east to west, and also decreasing from the surface layer to the deep soil layers. So what are the main sources of micro and nanoplastics in arable soils? We think that they have at least 8 different sources. Most of all is the plastic film residues. It's the use of the plastic films, mulching films, which is very quite often used in the agriculture production. And then is the sewage irrigation and also sludge utilization as fertilizers, and also the application of organic fertilizers. The fifth one is the atmospheric sedimentation, and also the surface runoff with microplastics, and also the landfill of plastic debris. And the eighth one is the illegal dumping and weathering of the tires on road. This is summarized in a review by our group. Our group have initiated 3 long-term field experiment to show how these different important pathways that could contribute to soil microplastic pollution. They are long-term application of pig manure, long-term application of sludge, and also a long-term mulching. So these experiments were conducted in different provinces of China. And then we use different methods to identify and quantify the microplastics in the soil, and to show their colors and particle size and types and composition, to calculate their abundance and to show their surface characterizations. And from the results, we found that the abundance of microplastics increased significantly with the mulching years, and the filmy microplastics, the film one, accumulated in the porough layer are estimated at 18.1 million items per hectare per year. That means that the mulching and the long-term mulching is a very important source of the microplastics in the soil. So when these plastics are in the soil, do they change or they don't change? We found that the combination of mechanical friction and the chemical oxidation or biological erosion results in the appearance of scratches, wrinkles, micropores, and cracks in the surface of microplastics. You can call the weathering or the aging of microplastics in the soil. You can see the surface morphology changes. Besides the physical-chemical changes on the surface of microplastics, the other one is the biofilm formation on the surface of MPs. The microbes could secrete the EPA's extracellular polymer substance to show the biofilms. In one of our experiments, we use the confocal laser scanning microscopy to show that with the time increasing, the biofilms increased. The identity of the biofilms increased significantly. The green fluorescent dye shows a living bacterial cell on the surface of a microplastic film. and the blue one shows their EPS, and the red dot shows a dead cell in the biofilms. And also we show the different microbacterial community abundance at a violent level among different areas of the microplastics in the soil. We found that it changes a lot. The main microbial community abundance changes with the different climate zones. And also we found that when the biofilm forms, it not only shows the microbial changes, it also can uptake or accumulate different pollutants such as heavy metals like copper, and also different organic pollutants. And moreover, the micro and nanoplastics in the soil, together with their coexistent pollutants, can be uptaken by plant roots. And with a cracked entry mold, and then it could be transferred to the upground of the soil. In one of our recent study, we quantified the amount of the micro and nanoplastics uptake and transport in plants with a rare earth labeled methods. And then this will lead to the food chain transfer, and then lead to the risks of human health. Recently, we write a review in the EAH journal to show the different risks. So, as we know, the microplastics and the nanoplastics in the soil may cause the risks for the environment and human health. Then how can we control? In China, we issued an action plan for plastic pollution control of the 14th five-year plan. The key point of this plan is to pay attention to three steps. The first one is the source control. That means we use less plastic materials and to recycle them. The second one is the recycling processes. Then we use different ways to recycle the industrial, the living, daily living, and the agricultural plastics in many ways, in different ways. And the last one is to develop different kinds of end-of-pipe treatment or the remediation technologies. But along the different technologies of decreasing or remediation techniques, one of the most emerging one is the microbiodegradation. It is an important technology to beat plastic pollution. Due to the full range of substance of microbes, their abundant application sceneries, and a high metabolic potential, and also low cost and consumption. So microbiodegradation is not only effective to organic or heavy metals, but also to the new pollutants like plastics. In 2016, a Japanese scientist found and a report in Science that a bacterium that can degrade and assimilate PET, it's a kind of non-degradable plastics. Drinking bottle or drinking water bottle is made of PET. They find this bacteria that can degrade this kind of PET plastics. And since that, different science from different countries have also identified a different kind of microbes that could degrade different plastic types such as PE, PVC, PP, PS, and PUR. So, but all these are non-biodegradable plastics. So, but recently China is trying to develop or to use a lot of biodegradable plastics or such as the biodegradable mulching films called PBAT. So my work is going to screen and identify a kind of strain that could degrade PBAT mulch films. So we found some we select some soil with mulching film residues from the field, and then we use PBAT, the mulching film, as the soil carbon source. Then we co-culture them and to bring some colonies, bacterial colonies. Within the solution, we found 9 PBAT degrading strains after preliminary screening. Then we re-screening of the PBAT degrading bacteria to inoculate the 9 different strains into the solution with only mineral source and carbon source free mediums. Now to check the growing curve of the different strains and to choose one which can grow very fast within the system with no other carbon source. After 40 days incubation, we found that the strain PB88 could degrade the PBAT films seriously, and it received a weight loss of 6.6% after 40 days. And then we select this strain as the target strain. We observed their colony morphology and their shape of the bacterium. It's a rod shape under the SEM. And also we checked the enzymes activity of this strain. We found that it is positive with urease, casein-ase, lipase, and some catalase. With this enzymes activity, we think that they could degrade the PBAT under this enzymes activity. And moreover, we did the molecular sequencing and the identification of the strain PB88. After the phylogenetic tree analysis, we found that this strain belongs to Bacillimonas species. When we look at the growth of this strain in the solution, we found that the OD600 growth increased very significantly after 60 or 80 days of the inoculation, and then the PBAT films shows very significant changes in their morphology, with many holes and cracks in the surface, and also the biofilms formation on the surface. And also we use the confocal laser scanning microscopy to show the composition of biofilms on the surface of the plastic film. We found that you can see also the green fluorescent dye shows the living cells. The red one shows the dead cells, and although the blue one shows the bacterial EPS. We found that the dynamic of biofilm composition changes after the strain inoculation, and with the increasing of incubation time, the living cells decrease, and while the dead cells increases. And also we check the hydrophobicity of PBAT. It decreased with the inoculation of the strain and also with the incubation time. After 6 months incubation, the PBAT film was seriously degraded or damaged, and it cannot even support the measurement of the water contact angle, the water drop. It cannot support the drop on the surface. So the hydrophobicity decreased significantly. And we also check the functional groups on the surface of the PBAT before and after the inoculation of the PBAT8 strain. We found that different carbon and the carbonyl and the ester groups were increased on the surface of the strain of the PBAT after inoculation of the strain. That is a signal of the bacterial degradation or bacterial oxidation. As we know, The main mechanisms of microplastic biodegradation is the enzymes activity. For the non-hydrolyzable and hydrolyzable plastics, the different enzymes were used during their biodegradation. So to identify the functional degrading enzyme genes in our strain is most important. Then we did the whole genome sequencing with the next generation sequencing technology. And we found that the strain has one chromosome and plasmid genes. Then with some omics analysis, we have found some different expressed genes that were in the strain before and after adding of PBAT as the carbon source. And now we are doing the further analysis of the genes that were involved in the degrading of the bacteria. So this is an ongoing work. So I'm not going to conclude with the PPTs today I'm sharing, but I just to say that I'm going to do more the deep work on the omics and the molecular analysis on this strain to show the detailed mechanisms that were involved in the degradation of PBAT by this strain. And hopefully I can show you some recent results in the next meeting, IPC meeting. Again, thanks to all the foundings and our group members, Professor Law and all my students. And also thanks to the organizers and our colleagues in our society. Thank you for your listening.
So thank you, Mr. Chu, for this presentation. I don't think we have time for question. And is he online? No. So maybe students could have a remark, comment on this presentation, on this content. No question, just a remark. No success. So
no, just an information. It's me, Greg. Oh. We have a French company. The name is Carbios, and they are working on the enzyme involved in the biodegradation of different plastic.
Thank you, Michel, for this precision. So let's move to the last presentation of this morning, and please warm welcomes to Isabel Churchill from the University of San Diego in USA, who's going to talk about a key point of our works, which is the marketing techniques to promote phytoremediation, awareness and acceptance. So Isabel, the floor is yours.
Sorry to cut you. Just one more thing. Isabel is a student presenter, so consider to filling your evaluation sheet.
Yeah, and 12 minutes for the presentation, and then 3 minutes of question. Isabel, the floor is yours.
Okay. Thank you so much for this opportunity. My presentation is going to be quite different from the rest, as it's focusing on marketing techniques to promote phytoremediation awareness so that it can be better understood globally and reach a wider audience. IPC is such an amazing event as it brings together people who share a technical excellence and passion for these phytotechnologies. However, the success of these technologies depends on the commercial application and adoption so that it is able to reach its maximum potential. The challenge is that these technologies are not well understood outside of areas such as IPC, so we need to do more to educate the benefits around this so it is able to reach a wider audience. In this presentation, I will share some of the techniques I used while interning for Intrinsic Environmental, which is a phytoremediation company. Their solutions were groundbreaking, but they were needed marketing to help reach a wider audience. My goal is to communicate phytotechnologies in a way that can be understood by all, to share the necessity that this technology brings to commercial companies faced with challenges of pollution, contamination, and government regulation. So let's get into it. So first, I'm going to tell you a bit about myself. I have an honors business degree in marketing with an emphasis in sustainability. I had the opportunity to work for Intrinsic as I loved what they did, and I thought it was so fascinating that you could use plants to clean up our environment. Going into this internship, I had no prior knowledge of phytoremediation, but I quickly learned and recognized that Intrinsic Environmental was not reaching their audience as well as they could. I recognized the importance of marketing this to the average consumer to make it more understandable, as many commercial enterprises are not aware of this technology, so how can they know the benefits of using it? It's the commercial application of this technology that's going to drive its success and have the greatest impact on helping better our planet. So I started by completely altering their marketing approach. So what Intrinsic Environmental does is endophyte-assisted phytoremediation, or endophyte-enhanced phytoremediation. So they have licensed these contaminant-specific microbes that they inoculate into the trees. And I like to explain this to my friends as it creates super trees, which feed off contamination as their food source. So they have them for oil and gas, TCE, PCE, RDX, and PCB, and these can also be combined for mixed waste sites. So this helps differentiate Intrinsic Environmental from regular phytoremediation, as these microbes are more efficient at cleaning up the contamination and help the trees have a higher survival rate. Some people who may know about phyto, but they need to be educated on the use of endophytes and how this can help assist in regular phytoremediation if people have had a prior experience where maybe a site was not able to survive. Here's highlighted 6 reasons why that I wanted to communicate to companies why they should use endophyte-assisted phytoremediation, as it is a lower cost compared to other traditional remediation methods. It reduces a carbon footprint, it increases land value, it's sustainable, regulator approved, and has a beneficial community impact as it helps restore environmental justice to communities. So now let's dive more into how I marketed phytoremediation. So first I started by identifying the problem, which is phytoremediation is understood well by very few people, and even those in relevant industries may be unfamiliar with the term or the benefits that it can bring to commercial enterprises. So I started by identifying our target audience, which is anyone or anywhere that is dealing with contaminated land and groundwater. And this could be businesses involved in oil and gas, waste management, reforestation projects, air bases. There's lots of examples. And these organizations need to know that phytoremediation can help them solve this problem sustainably, as well as help them meet environmental and legal obligations. So my position for this approach is based around the benefits and raising awareness for this solution. I redesigned Intrinsic Environmental's website to make it more user-friendly and understandable to all. I ensured that we showed up at the top when people searched for sustainable environmental solutions when dealing with contamination, as well as I developed training programs for our new employees, which educated them on different environmental laws and regulations in government agencies. I assisted in creating one-page briefs that we used at conferences that highlighted the uses of endophyte-assisted phytoremediation in mine sites, petroleum remediation, brackish site restoration, and many others. And we had these available at conferences to showcase to people examples of how they could use endophyte-assisted phyto. So having an accessible website and good marketing content is crucial, but so is leveraging this through social media. And I specifically focused on LinkedIn. So I know everyone hopefully has a LinkedIn in here, but very few people actually leverage their LinkedIn effectively. So marketing through LinkedIn was crucial to my approach, and I educated our team on how to effectively use their LinkedIn, and I ensured that everyone was active on their profiles and that they matched the vision of the company. So people tend to trust and respect a company when their employees are active and passionate about what they are doing and what they are working for. So when creating content, my goal was to bridge this gap between these complex environmental solutions and public engagement. I created posts that were easy to understand to the common person who had no knowledge of phytoremediation. I made this available to the intrinsic team to post, as well as posted it on the company's LinkedIn page. It is so crucial that you guys share the importance of your science online, because it may get written in journals, but journals alone will not spread the word globally. So leveraging through LinkedIn will help you reach a wider audience. Here's two examples of some posts that I created. The first one is a before and after of showing a site that was contaminated with diesel fuel and benzene. And the after is shows the beautiful restoration that we did with our trees to clean up that contamination. Previously, this site had a failed soil vapor extraction as well as a pump and treat system. So that's why we were brought in. And we stopped the intrusion from getting into the community. And now it is a park. So this is a great example that visually shows how phyto can help restore and show biodiversity to communities. And the second one is an infographic about wildfire restoration. And this is really crucial for where Intrinsic Environmental is located, as it is in California, where we have lots of wildfires. So it's good to educate people on how phyto can be used to help restore and regenerate post-wildfire landscapes. Okay, so this is the last topic that I will talk about in my marketing approach. So I aligned phytoremediation with how it supports companies in meeting ESG and sustainable development goals. I know you all probably know what sustainable development goals are. They're outlined by the United Nations, and there's 17 of them that support businesses' worldwide commitment to sustainability. And companies today are having a much greater emphasis on being more sustainable and having to comply with with strict environmental laws globally. So implementing phyto offers a big incentive for companies to meet these goals. As there's, and they can be seen, having a strong ESG and SDG, and meeting SDG goals can help a company be seen as a leader in sustainability. For example, Shell, which is a very large petroleum company that most of us probably know, has a whole entire page on their website dedicated to the Sustainable Development Goals and which ones they are working towards meeting and how they are doing this. So the goal with this, so for goal 13 could be an example of phyto helping meet this goal, which is climate action, as it reduces greenhouse gas emissions compared to other traditional remediation methods such as dig and haul. And then goal 15, which is life on land, phyto helps restore ecosystems by removing toxic soil and helping companies protect their environment. So Overall, my goal with this marketing strategy is to drive corporate adoption by positioning phytoremediation as not only an environmentally sound solution, but also a profitable and compliant strategy for a company's long-term sustainability. Okay, so I know this has been a pretty short presentation, but I hope that I've been able to share with you the importance of marketing phytotechnologies and the benefits of creating a greater awareness and some of the methods that I used while working with Intrinsic Environmental, which helped them better reach a wider audience. The goal is to be able to talk to prospective clients about phytoremediation in a way that is easy to understand and gets them just as excited about these technologies as we are. So, in conclusion, that education in a way that is easy to understand and leveraging this through social media platforms to reach a global audience, and then aligning with universal practices to implement an incentive for people to want to use these technologies. It's so imperative that more people understand these technologies, and I encourage every single one of you to work loudly on LinkedIn and share your technologies and experiences in a manner which everyone can understand. We need to bridge the gap between these complex environmental solutions and the common person so they are able to understand. So feel free to reach out to me if you would like my assistance and connect with me on LinkedIn. And if anyone in this room doesn't have a LinkedIn, you better make one today.
Thank you very much, Isabel, for this very nice presentation. Does anyone, students first, have question for Isabel? Come on, students.
How many students are on LinkedIn? Who's on LinkedIn?
Who's on LinkedIn?
All the students? Some students still need to do that? Okay, yeah, look, come on, link in.
So, no question? Well, please.
Thank you, Isabel, for the wonderful presentation, very useful. I have a question. How can I describe what I do for phytoremediation without being reductive to my production and making it understand to lay people? Because what I feel when I try to be understood by the normal person is that I reduce my work to something hilarious. Oh, plants take up the pollutants. Wow, that's cool. No, I don't want to do that. My work is much more than that.
Yes, definitely your work and everything that you've put into it is so important. But so maybe if you were posting on LinkedIn about your work, you could highlight the uses for it and then have a link to all the research and everything super scientific in that space, so people, if they want to learn more about the science, can look into that. So it's like best of both worlds.
Okay, thank you.
Thank you very much. I think we have time for last question, Azam.
Thank you for the very interesting and informative. I do have a quick question. You may have interacted with some clients, so you may know the answer or not. How would you deal with clients that are a bit difficult? So I understand LinkedIn is going to have a lot of information. You put your information out, people may have time to read or not, so you try. So that's the part is just providing information and outreach. But beside that, as far as the communication and outreach, the goal was trying to convince and inform the clients, and then let's put in the parentheses maybe difficult clients, that if you go with phytoremediation, if you have that gas station that's contaminated, you go with us, it's going to be aesthetically beneficial, it's going to be cost effective, and all of those things. How do you deal with them? And they don't have time to go to LinkedIn, they are not going to do that. Verbally, how would you do that?
Yeah, so usually if we're dealing with a difficult client who is skeptical about using phytoremediation, we can show them examples of past sites that are similar to what they have dealt with, and how successful it has been, and the methods that we've used to implement that. So it kind of is a good example, so they trust that this will work as well as it did prior.
Thank you very much, Isabel. We can thank her again. And so that ends this session. So maybe we could thank again all the speakers of this session. Very interesting session. And now I invite you to go to the lunch break. Comment? No. Thank you very much.
The concluding keynote speaker, and I invite Dr. Jason White to introduce the keynote speaker, please. Jason.
Well, thanks, Om. So it's both an honor and a pleasure for me to introduce Professor Baoshan Zheng, not just because he's an exceptional scientist, which you'll hear about over the next 35 minutes, but actually because he's one of my closest collaborators and friends. I promised him I would keep this brief because to adequately introduce him, I'd need 10 PowerPoint slides in 15 minutes, so we won't do that. But his current position, he's director of the Stockbridge School of Agriculture and a university distinguished professor. His general area of expertise is in environmental and soil chemistry. He's received his bachelor's from Heilongjiang Baoyu Agricultural University in agronomy, with then a master's in soil chemistry and mineralogy from University of Alberta, and a PhD from the same institution in environmental and soil chemistry. He then actually did a 2-year postdoc at the Connecticut Agricultural Experiment Station and ended up leaving just about a year before I left. So we didn't overlap, but we became collaborators and friends shortly thereafter. Just to highlight a few of his achievements, he's been a highly cited researcher since 2014, which is probably pretty close to the record for anybody. He's been ranked as among the best scientists in environmental sciences by Research.com. He's published nearly 900 papers. His H-index is an unbelievable 152. He's got more than 97,000 citations, and his I-10 index is 859. So, but separate from all of that, as I mentioned, we've worked together for many years. He's among the best mentors of students that I've ever seen, and just a pleasure to collaborate with him and anybody who has ever worked with him. So, Baoshan, I said I'd keep it short, so I kept it short.
Good afternoon. Thank you, Jason, for that kind word. I give all the credit to the postdocs, students, and visiting scientists. I could not do all that work by myself. First of all, I'd like to thank Om, Josh, for organizing such a fantastic conference, and also providing me this opportunity to speak. And also, I really, from my bottom of my heart, to thank all those unsung heroes. Those are the people who with the yellow badge. Those people really help us to have this successful conference, make it very smooth. People with, thank you. Slide, registration, give directions, you name it. Without them, we would not have such a smooth, successful conference. Thank you again. So the title is given by Om. I work on microplastics, but I never work plastic-related human health issues. I said, okay, that's an assignment. Just like your professor give you assignment, you have to do it. So I dig into the literatures, and last few slides is indeed will be on human health. So that's the overview for my talk. Before I get into my talk, I just would like you to know I'm not trying to scare you here, okay? But definitely after my presentation, the take-home message is you're aware of the issue with plastic waste. The series of pollution from microplastics, that's all I need you to know. Let's talk about plastic production. So that's the current data I have from 2009 to 2020. The whole world per year produced 227 million tons per year to 367 million tons per year. If you talk about the distribution, Asia, that's including China, 52%. China alone, 32%. Japan, such a small country, that's 3%. India, likely will be 12, 13%. I Googled a couple of nights ago. India produced 20 million tons per year. 20 million tons per year. The recycle percentage is only 7 to 10%. 10% is maximum. Where the rest of that 20 million ton plastic go? Most likely end up in the landfill site and also the environment on the street. So the slide on my right, maybe both sides, you see the few major type of polymers: polypropylene, high density, low density polyethylene. That's the in agriculture, not here. In a cold place, you have plastic mulch, more or less, it's polyethylene. I do not like to see those pictures. Unfortunately, that's the reality. You see piles of plastics get into the environment. Look at this number, almost 8.4 billion tons accumulated in the environment. Every single year, you got 4 to 12 million tons per year get into the ocean. That's the underestimate. You kill, not you, everyone contribute to this plastic pollution. Kill 100,000 marine mammals, marine lives per year. Just 2 years ago, it's a UN Environmental Assembly get together in one of African countries. I forgot exactly which country. They try to come up an international legal binding agreement by this year. Unfortunately, I haven't seen anything happen yet. 2 months left. They try to come up with an agreement like carbon dioxide emission. Nothing happened yet. I'm sure a lot of political struggles between countries. Just 2020, COVID pandemic, that year alone, you have 1.56 billion disposable masks get into ocean. You have much higher number get into landfill site, on the street. Plastic, the scientists long time ago, 30 years, 50 years ago, when they make plastic product, they have a goal to make sure the plastic lasts forever. That's their goal. So that's why those disposable masks, they make basically 3 layers of polypropylene. Once it gets into the environment, easily last 300 years, 500 years. Because those polymers, highly hydrophobic, very difficult to degrade, hydrophobic materials. What's microplastics? So in 2004, long time ago, that first introduced microplastics. Anything less than 5 millimeters, not in micrometer scale, millimeters, we call it microplastics. Bigger than that, called big piece, macroplastic pieces. Then in the nanoplastic just lasts probably 5 years or so. This term is coined, nanoplastic. There's still debate which nanoplastics. I used to study engineered nanomaterials with my collaborators, Jason White and also Ohm. In nanotechnology, nanoparticles are 1 to 100 nanometers, nanomaterials, at least in one dimension, that's for sure. When you're dealing with plastics in the environment, that's very difficult. It's very difficult to collect, enrich the nanoplastics in truly nanometer scale, like 1 to 100 nanometers. In our lab, we've been spent 5 years to do so. It's time consuming, student complaints, I complaints. So that's why in my lab, we use 1 to 1,000 micrometers. So often we're dealing with plastic particles 500 nanometers above, because that's technical difficulties, research limitations, analytical limitations. We get a sample from blood, plants, soil. It's not trivial to get nanoplastics. In general, we have two types of microplastics. Primary microplastics, that's the manufacturer. They make this particle like 1 micrometers, few micrometers. Like for example, in cosmetic industry, as a vector, as a carrier. Those eventually will get into the category of the one I mentioned, secondary microplastics. That's like a mineral. We have primary minerals, secondary minerals. Primary plastics eventually get to secondary plastics through resin. That's what we worry about. That's abundant in the environment. Now, what's the distribution? Where they are of those microplastics? We have two talks, unfortunately, through remote. You already know microplastic in the soil. Then, if the microplastic in the environment, are they toxic? What's the toxic mechanisms? If they're toxic, do they pose any risk to us? Let me talk about the distribution. Few slides. Try to convince you they are everywhere. This is not from our work. This is from the literatures. I have the citation right here. That's from way south, from most south than here. That's Antarctic pole. You see the polystyrene. You see the polystyrene film. Once it breaks down, make microplastic particles, the poor marine organisms you see in the body. I'm sure they don't feel comfortable. If we eat anything get into my gut, that's not very comfortable at all. Then we go north, Antarctic, the Arctic, the ice and some snow samples. Every single sample, you found microplastics. Over here on my right, you see that type of polymers you have. I have a number here per liter. The ice, snow, you melt it, become 1 liter. You talk about 10,000, 14,000 plastic particles in 1 liter. Do you want to drink that water with that many particles? I don't. But that's an underestimated number. Simply because, as I mentioned earlier, some nanoplastics, they are unable to detect. They are unable to collect. Real number will be a lot higher. Then we go to deepest place on Earth. Everybody knows that's Mariana Trench. We talk about 10 kilometers down. Sure enough, not easy. That's not my group. Some other groups, they spend time, special vessel, go down there, get the sediment, get water. You get all type of microplastic particles in there. How do they get there? Some aggregations, some get on some benthic organism, but exact processes we don't know. I hope some young, talented man here will get on, because this is pretty close to the ocean here. So we don't know how to get there. Then we go to somewhere not too far. It's highest place on Earth, Mount Everest. You see the fibers on my right? It's likely to plastic fibers coming from mountain climbers. However, we just have recent data. We have in high elevation, the animals there, we collect their poop. Sounds gross. We do study like that as in there. How do the particles get in there? Atmospheric transportation. Just any contaminant, atmospheric transport, like Arctic, Antarctic. Now, human activity is really low. United States, we have beautiful national parks. Those very remote. On tourists there, you sample the air in the park. 98% of atmospheric sample, you have nanoplastic, microplastics, likely through urban center. That's the microplastic produced through air, transport to those remote parks. Couple of slides, one dealing with the marine system, marine sediment, marine sample, a few data in India, every single places we found microplastic particles. That the concentration can vary by 4 orders of magnitude depending human activities. What about fresh water? Fresh water, lakes, rivers, you got quite a few data points right here in India. Sure enough, you have quite a bit of particles. Some places have more particles than others. Last few slides, I just convince you. I have a lot of data maps. I hope you have the idea. Microplastic particles almost in every single environmental media. Now, once you get in environment, what's the processes? To sum it up, probably just 3 main processes. First, that's my previous life, dealing with absorption, desorption of organic chemicals. Same here. So those yellow ones indicate heavy metals, organic chemicals. They can absorb on microplastic particles, depending the properties, the surface function groups. How many of them on the plastic particles? Sorption, desorption. Those particles can add a carrier. Concentration of particular contaminant in water not too high, but those plastic particles can concentrate those contaminants. Then you have aquatic organism, inject them, all of a sudden you download these nasty chemicals in their gut, then you have toxicity issues. Not only particles, but also chemical, the particle carry weight. Now, any particles, that's cloudochemistry. You can go through aggregation and dispersion. So as you can imagine, individual particles, aggregate particles behave totally different. Toxicity, transport, ability to cross biological membranes, once they get into organisms, they're all different. Of course, we have degradation, weathering. Some people see aging from small particles, from a bottle like that, in the environment, then break pieces, then produce smaller particles. Smaller particles need more attention. Again, from my studies, many other dealing with engineered nanoparticles. Don't worry about this basic slide. They have this few most polymer type studied by scientists. Then you have different organic contaminants like PCBs, PAH, antibiotics, many heavy metals. Basically, that's wonderful. Like other scientists, we have to start from simple system. Unfortunately, those are lab work without considering the environmental relevant microplastics, where you have functional groups, where you have biofilms, where you have a corona on the surface. Those ones just pure particles, pristine particles. Give us enough information, but those studies do not represent what's happening in the environment. For some young scientists here, particularly graduate students, you want to study plastic particles, try to weather them. Then you carry on your study. This is our own work we published a few years ago dealing with straight antibiotics. So those first group of mechanisms are already proven. Those one, few here, in our lab, we'll try to investigate, are those commonly bound for active carbon, biochar, are they responsible for absorption of antibiotics? So far, we don't know yet. Only one thing we can see, this negative charge since 100 bond can play a role for absorption of antibiotics by microplastic particles, weathered microplastic. First three, we still doing some work to verify if they can form, bond microplastic and antibiotics. Again, in the lab, you beautifully study all the pristine nanoparticles, spherical. Unfortunately, I think my friend Jason said earlier, soil is a black box. Not as black as he tried to envision, but we do understand certain minerals, clean minerals, and oxide. I think yesterday some speaker talked about how the montmorillonite and kaolinite affect microplastic uptake by plants. One thing probably wrong, all his polystyrene particles carry positive charge. He mentioned montmorillonite have big interactions, reduce plant uptake due to aggregation. From our data and others, you can see that goethite, magnetite, only the positive charged soil mineral particles, they can hit to aggregate with all type of microplastic particles with negative charge. Go through weathering, most plastic particles carry negative charge. You see kaolinite and montmorillonite, they don't do very much because they're highly negative charged in most of soil. Unless it's solely acidic soil, kaolinite can carry positive charge. So when you study microplastic particles, beautifully you can do in the lab to do heteroaggregation. We do that too. I'm not criticizing people doing that, but you have to be realistic. You talk about their behavior in the environment. You got to study heteroaggregation. You got to study organic matter, dissolved organic matter. You have to study biofilm. Weathering, that's a UV. We have this solar light. Visible light, we don't do very much. They're not highly energetic. But UV light, that's you and I, when we go to beach, we put sunscreen, protect UV light, not visible light. Go through UV light, then you can see in ocean water, weathering, it's more intense under UV radiation than fresh water. You have chloride there. And the UV radiation, chloride can produce different type of radicals in addition from water, like hydrogen peroxide, superoxygen. That's why in ocean water, you weather more than fresh water. They did not give an explanation. That's not my study. That's my explanation. This is our own work. We published a few months in Nature Nanotechnology. UV light, that's the most powerful way to produce microplastic and nanoplastics. Microbial, right now, you talk about microbial degradation. Early this morning, that's still minimum. Unless you train the bacteria. Again, they work beautifully in the lab. Once you get in the soil, you have biofilm. You have all type of microbes, bacteria, fungi. After compete, the one you selected, enriched in the lab. That's why the paper published in Science by Japanese group to degrade the polymer PET. It's, after that, almost like a light disappeared. If that's so powerful, we already have industry to do that now. I don't see any industry scale. Michelle mentioned there's a company in France that's doing synthesized enzyme to do the degradation. I wish them luck. So far, no industry scale to do the degradation, to get rid of the plastic waste in general. So some animals, essentially it's a mechanic degradation. They can, just like us, we chew our fur, we just have a tasty lunch. We chew them first, we don't just swallow them. Some, there's only 0.5 millimeter rotifers in any water, marine water, fresh water. You can see, I don't know, I have this video here, right here. You see how they have the chewing apparatus to break down. You see the plastic particles going in that chewing apparatus, get into their gut. During that chewing process, you make so many smaller particles. We use fluorescence labeled polymers. You can see so many small pieces. That's enough I'm going to go to the next slide. Oh, not let me do that. Okay, now we know different ways to produce microplastic particles, nanoplastic particles. Then they have, I mentioned earlier, through adsorption, microplastic particles can act as a vector. This transient host effect get into organism system. Not only particle toxic, the particles absorb contaminant. And also, whenever you make plastics, in the plastic you can have 100 other additives, also other chemicals. Those can release in the organisms. This is the Phytotech conference. I got to show a few slides with the plant. This is our own work, published 4 years ago in Nature Nanotechnology. Arabidopsis. So you see control, positive charge, negative charge, polystyrene, nanoplastics clearly get into the roots. This group data, not our data, that's my friend. I think your friend too, Yuming Luo from Nanjing. See, weight is root, stem, leaves. You get in there. Now, you may say, who cares? We don't eat leaves. We don't eat stem as a weight. What about the lattice? By the way, I love lattice. To get into the lattice. Not just ICM image. We don't know quantity. Then the same group, they use this special rare earth element, European. That's in the lanthanide group, in the periodic table. Two rows underneath the periodic table. The first row, you find lanthanide theory. They detect, this milligram per kilogram, we talk about root, the lattice, can be 10 milligram per kilogram. That's quite a bit there. This is our own work. You can see control, negative charged particles, positive charged particles, retarded the plant root development. This is our own work again. Jason is on this paper. We published 4 months ago in ACS Nano. We root soil exposed nanoplastics. You can get, you see, roots, shoots, leaf, and fruit, not tomato fruit. If you incubate them long enough, I'm sure in fruit will be more. The one on my right, this another paper. This is doing foliar exposure, just like mimic atmospheric deposition on the leaves. Sure enough, those microplastics affect chlorophyll content, reduced biomass, root, and also leaves. Then you change the morphology of the leaves. Interestingly, just like we studied copper oxide nanoparticles, they can get the leaves, get in the stem, get in the roots. I'm not a plant physiology person. Someone need to figure out why plastic particles can go through phloem in there. I can see nutrient, copper oxide nanoparticles, they need copper. Plants do not need plastics. Why? What on earth they go through these difficulties getting to the roots? Not relate to you and me now. This paper, I don't know how accurate it is. You and me and IDOT consume 5 gram plastic a week, equivalent to a plastic credit card. Goes through inhalation, ingestion, dermal not a big issue, so we now go through that. This is our own study. We published 2022. This is baby bottles. You don't use the baby bottle anymore, but your parents used the baby bottle to feed you before sometime in your life. We use the nipples. We go through WHO standard recommendation for sterilization. Steam, go simple process. You can produce 0.6 million those particles through 1 year of a baby infant. Again, that's underestimate because our instrument can only detect 0.6 micrometer. Anything below that, our instrument is unable to detect. So that's why I'm saying this number is really underestimate. During the COVID, we cannot eat in the restaurant. Now we have a habit, take out. We studied different type of take out plastic particle. This is published 3 months ago. Goes through the same microwave, steam heating. You see the particles coming out. Our lab, the chemistry lab, we are not allowed to use animal. We don't have a permit, but we can use human cell lines. This is human cells. We buy them, we do the incubation. Sure enough, the toxic human cells. We cannot extrapolate to human, even to animal, but at least the potential toxic to you. This is a group from medical doctors that a couple of years ago in environmental science and technology, sounds gross. This group of people collect human stools. They isolate the plastic particles. You see different shape, all type of shapes in there. I want to draw you, I don't know if you can see that. There's a correlation between fecal microplastic concentration and inflammatory bowel disease. Many people, adults, have this inflammatory bowel disease. Maybe there's a connection, but I'm not now saying that's the cause. But at least something you need to think about. This is our own work a few years ago now. We use good oil, olive oil from Spain, And we go through different type of particles. Clearly you can see all particles reduce digestion of olive oil in simulated gut. But we cannot use people, we use simulated system. New England Journal of Medicine, one of the best medical journal. That's a group of people from Italy. Then Harvard medical scientists. You can look at it. They have 350, 400 heart disease patients. Medical doctors have access to those plug in people's blood vessel. Their conclusion, you can Google this paper. On New York Times, many news media. The people with microplastic particles in their blood vessel, they likely suffer heart attack and stroke. They follow those patients for 3 years. Those patients with plastics, they put here, likely to die over the next 3 years than the people without microplastic. This is not our work. Go check that out. It's published in March. This paper, I still have some time, but I try to finish soon. You can ask some questions. These people have not even published. It's preprint through NIH website because those people got funded from US, the National Institute of Health. That group of scientists, toxicologists, forensic scientists. Of course, you cannot use real people. They're real people. They're dead. Okay? They sample the dead people, kidney, liver, brain. So for some reason, brain had higher plastic concentration than liver and kidney. Jason is the toxicologist. Often we have pesticide, antibiotics. Those go to our liver first. We often find like a fish, like a mass. Often the liver has higher concentration than other organs. Microplastic behave differently. There's here microplastic and nanoplastics. For some reason, for those people, brain have higher. Then from 16 to 24, every single organ, microplastic particle concentration increased. That indicates we expose more of the particles over the next few years. Animal studies, many animal studies, not us, in literature, they link microplastic particles to low fertility of animals, mice. That's concerning. Worse, cancers, disruptive endocrine immune system. That impaired learning ability, the memory. My memory declining. I don't know if it's due to microplastic particles or due to aging. But anyway, so that from animal studies. Look at this. The same study I showed you in previous slide. That scared me the most. They have 12 brain samples. Those people have dementia, like Alzheimer, memory issues. Those people in their brain, microplastics, nanoplastics, up to 10 times higher than other group of people died from other reason. Without microplastic, 10 times. And that relationship between people with dementia and microplastics, I don't know. But that's the paper will be published soon. This is unpublished, preprint. In US, we don't have any governmental standard to plastic particle in water and food. PFAS, EPA just have standard for 4 6 in drinking water. Now the EPA try to come up with some guidelines how to measure them in water and other samples. I'm going to quickly conclude: microplastic everywhere. In my blood, for sure. Absorption, aggregation, degradation is the major process. And also, microplastics can ingest by all type of organisms. They can make them smaller, like earthworm. Study on earthworm, I didn't show you that the slow growth and also DNA damage in earthworm definitely can get into our system through ingestion and inhalation, through, like, let's say, microwave your food in plastic containers. As I said earlier, all the work is done by my students, postdocs, collaborators like Om and Jason, and many visiting scientists. I intentionally talk fast. Then I have something at least 7 minutes, because we started something like 2:32, 33 like that. So I stop there. I'd be happy to address any questions. I may not answer your questions, but I attempt to address your questions. Again, I'm not trying to scare you. I try to raise the awareness, the issue with plastic waste. Thank you.
You want me to do it? Okay. Take some questions. Maybe students first. I got a lot of students sitting behind me. Well, we'll let Michelle start us off then.
In the newspaper, I read that we have concern about the microplastic in the mineral water, especially due to the caps. Do you agree about that?
Yes, I do agree. We studied this too. In our lab, we study not only the bottled water, we have tap water, we also study different chemical agents like sodium chloride, sodium hydroxide, because you use those acid base to digest the food, oysters, to isolate particles. Plastic particles definitely from the cap, from the interior wall, all depend how you handle it. If you're in your trunk, like today in your trunk, temperature can go to 35, 36, warm heat, you produce more microplastic particles. You put it in one place, don't agit too much, its particle it's much lower than you do the agitations. The type like Amherst supposed to be really have clean water. We cannot go to people's house to get tap water. We go to public libraries. We go to public schools. In our research lab, we collect those tap water. Sure enough, there are in those tap waters. I try to go to water utility. They are attitude almost like immigration officers. They are not very friendly. Get out. So I could not get any sample from water utility.
Elena.
Thank you for this wonderful, relaxing overview. I wonder, the epidemiological study, like you mentioned, 12 people with dementia, etc., considering the heterogeneity of the particles, do you think they can be safely attributed to the plastic itself, or more to the, like you said, all the contaminants they are carrying, all the heterogeneity, all the because it reminds me of the particulate, the atmospheric particulate. You have effect on inflammation, DNA, blah, 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 but maybe they are a concurrency of different pollutants.
Very good question and very good comment. I agree with you. I don't think plastic particle is the cause, reason for the people with dementia. Probably one of the factors. Like particles can be toxic, just like PM 2.5 get in your lung. They do not get out. They cause cancers. But smaller one get in the brain. And also the chemicals in those particles they can release. plus other issues. The question to me, how those plastic particles cross blood and brain, that membrane, that's somebody needs to study that. If you can stop there, would be good. So I don't know how that particles cross that membrane. And how the particles get into our blood. That I can speculate, because with all the particles get in our gut, get in intestine, they can go through that epicellular cell liner, then get into the blood. But another contribution, I'm not trying to scare you, during the COVID, I tried to get the IV bag, like a lot of people in the hospital. I tried to get the IV bag. I'm not a medical doctor. I'm not licensed to get any. You think about it, when I was a child, all the IV operation is glass bottle. I don't know if you recall that, some older ones. So that glass bottle, but now at every single hospital, because so easy, so convenient, you can buy already made, company shipped to you, IV bag. I went to an animal doctor, he gave me two. Sure enough, a lot of plastic particles. But I don't know, the animal hospital same, I'm sure it's different qualities. But if any one of you can get those IV back to me, I would love to study, because that directly into your blood.
Yes, sir. One question from us.
Yes, go ahead.
We have some plastics originating from bacteria also. Those plastics, are they that much harmful?
So people call it bioplastics.
Bioplastics, yeah.
But those ones compare the abundance, the quantity of human-made plastics, they're not big issue. But the bioplastics, bacteria, fungus, again, I'm not a microbiologist, they have a way to dealing with those ones. But the one synthetic plastic, they only have something like 70 years old or so. The microbes, fungus, they have not evolved to handle this yet. So most microbes, fungus, they can attack polar compound. So at least as far as I know, they're quite different.
Okay, thank you.
One there.
Yeah, you have shown some microplastics getting inside organs, but when they get into fibrillated arteries, are they known to cause blockage?
I don't know. The microplastic causes the blockage. Oh, like this plaque in the blood vessel. Maybe due to some cholesterol that they stuck there. Or the microplastic actually have to form that plaque. That part, I don't know. But that's an excellent question.
Because they need to have a minimum size to block the fibrillated arteries.
Mm-hmm. Yeah. I agree. I don't know. It's simply due to the plaque stop there, or the particles facilitate to form that plaque. That part I don't know. But a very good question.
Sir, excuse me. Is there any provision to measure this microplastic content in our human body? Is there any quantifying method?
So as I mentioned earlier, right now every single lab have their own procedures. We don't have any universally accepted procedure. Last year I was invited to go to Washington DC to give a talk. I said, let's cover some kind of standard. I got criticized. No way you can have some kind of standard procedures. You can see accepted by different groups. Don't call it standard procedure. So very difficult to at least right now to have a standard procedure. Every single lab have different procedures. That's the problem too, in terms of the inter-lab comparisons. I don't want to take too much time. I leave the time for Jason. You control the time, not me though.
How about one more question, which I will ask. So one of the things we know about engineered nanoparticles is that when they get in biological systems, biocorona forms, which significantly influences fate and distribution. You think the same thing happens for plastics, nanoplastics, microplastics?
That's I put right here. I think that definitely need to be studied. Yes, they do form corona, protein corona. They do form this type of things. So that probably, I could be totally wrong. I speculate. They form this protein corona, then get cross the membranes. They can fool the membrane. If it's pure plastic, they may not be able to. Mm-mm. But again, that's pure speculation. That's not. Well,
no, I completely agree.
Very, very short, Jason. Do we have, for other contaminants, adaptation of the biological organism to microplastic? I mean, adaptation of the population.
I don't know. That's a good question.
Good question for the students.
Yeah. And if I may elaborate, like PFAS, we called wonder chemicals, forever chemicals. Like 50 years ago, nobody cared. So we accumulate PFAS in our body. Every single of you will have PFAS in your blood. We are not adapting to them. In US, we have so much lawsuit dealing with low fertility, abnormal functions. So that's a huge lawsuit dealing with PFAS. I don't think we are adapting PFAS yet. Are we eventually adapt to plastic particles? If we do, great. If not, we need to do something. Thank you.
Thank you, Dr. Jing, for the excellent talk and creating and generating more interest for the young scientist students to pursue this research in the microplastic focus to solve the global challenges. Thank you very much. So now, yes, Bausen, can you please come back? So we have a token of appreciation for you for your excellent talk. Wait a minute, get your photographer, please. Where is the photographer? Take the photograph, please. So now the next session is Phyto Scholars and Career session. And this is meant for mainly the students. And there will be 3 moderators, Dr. Liz Rylett, Dr. Azam Noori, and Dr. Jason White. They will moderate this students and Phyto Scholars career session. And it's meant for mainly for students. All professors and all these, please go and have conversation outside so that students can freely talk and discuss without the fear of what professor will think. So all professors and scientists, please, and advisors, please get out and enjoy for half an hour outside until they have the conversation here. Thank you. And all students, please stay here. And any students outside, please bring them here. They will be talking about career options, job prospects in different research areas. Thank you very much.
It's an opportunity for academic networking, very prestigious academic networking opportunity for you outside. Thank you.
And I'm also out too.
Is this your presentation?
Yeah, thanks. I just wrote it. Thanks.
Something happened.
What happens is it has extended your screen. Where is your screen extended?
Who extended it?
I don't know who has done it.
No, it's not extended. Let me do one thing. Control Alt where's the delete? Yeah.
It doesn't like me.
Yeah, I just came to see what's here, but it's so nice hand actually.
It's okay.
It was formed. Just go off and on again. Restart. I mean, the good part of we will remove the audio, right? No, audio is not recorded. Zoom is not connected, right?
I can start while you get it.
Okay.
Go to Kerala presentations and then date modifier should bring it to the top.
Okay.
Student career support. Thank you. This is a short one.
So I will put it there on the session.
Thank you. Yeah.
You can get it from there.
Okay, I'll try and we'll take 20 then. 20 will be plenty. Okay. Right, get out. Okay, everybody, we're getting our presentation up. Can you all raise your hand if you have asked a question at this conference, please? Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, and I'm 10. I make that. Okay, and now if you are female, raise your hand. Come on, if you're not sure, raise your hand as well. We'll count that. Females, are you female? Female, yeah, come on. Okay, right. Did you notice how many hands were up that were male for the first question? Okay, I counted 7 of the 10 were male asking a question at the conference. How many women are here? Way, way more. Come on, women, what are you doing? Why are you letting these men ask these questions? You should be asking the questions. Why are you already slipping behind? Ask some questions. We're getting angry now. Okay, why should you ask questions? Because, number 1, you're really interested. Yeah, I want to know. Huh? Number 2, the speaker wants you to ask a question. I don't want to stand here and give a presentation that I wrote in another country. I came on an aeroplane, I stood here, I presented the work that I've sweated over in the laboratory, and then not a single question. I feel bad. Don't leave anybody in that position. Always have a question. Okay, then I feel good. Thank you. You feel good. Hey, asked a question. Okay, everybody's good. The audience is good because the audience got nervous. Oh God, no one's asking a question. Nobody likes a silence, do they? It's uncomfortable. Keep everyone happy, yeah? So you ask a question, that's good, everybody's happy. And then question the science. Okay, don't be rude, be polite, but, is it good? Have you got any suggestions? What could happen? So questions just ask. Nobody's been shot for asking a question. I don't think anybody's been at all hurt. In fact, I suspect they've all survived. Did you survive your question? Those that yeah, they're still here. They're fine. Do you feel good? Yeah, you feel good. Yeah? Okay, it's a win-win. Ask questions. Come on. Right. Okay. What have we got the graph behind? Hang on. Let's get, yeah. All right. So we're the big penguins, and you guys are the fluffy penguins. That's the idea. Oh. Okay. Asking questions. We're done. Right. The value of a mentor. Azam, would you like to speak?
Sure. Is it a little awkward? I go off your slide. I'm sorry. I'm just going off my memory, but use your links. Mentorship and having your mentors. Being a trainable person is important. So somebody who is your mentor, is your professor, is helping you, has years of experience on their backpack. It's good to understand what they are trying to convey and what they are trying to give you. And you be a trainable person to give that message is putting you already a step ahead. So trying to, even if you feel I'm not receiving that from my own mentor, trying to networking and going with somebody else, asking them for help, that's valuable skill. I would say as far as, soft skills, hard skills that we all need to build, the ability to network and go find that good mentor for yourself, that's very important. So if you have any comment to add.
Sure. So I guess I would add that really science doesn't work without mentorship. I mean, that's the only way a field of any endeavor is going to advance. But it's also a two-way street. I mean, the older you get, and when you get really old like me, you spend more of your time as a mentor. But all of my people who work with me and work for me, they're mentoring me as well. I mean, we discuss what research we should do, and then they go do it, and then they tell me why I was wrong, and we should have done something else. So it's not just a one-way street. It's about communication. And if your advisor thinks differently, get another advisor.
Okay, good. Thanks. I'm just going to back up to the questions, just to give you some examples of questions you can ask. You can say, well, you did that in copper? Why did you choose copper? Could you use zinc? Did you try with zinc? You could ask, why did you choose that plant species? Have you looked in another plant species? A real old chestnut, the supervisor that wakes up on the back row and is asked to ask a question. They go, what is the future direction of your research? Because you can ask that of any scientist, yeah? So these are questions that the speaker's grateful. They know it's a fairly easy question, but they're grateful, the audience is grateful, you're grateful. There's so many easy questions. Even if you have not understood the science, you think, they're working on carrots. Have you looked in apples? I haven't even seen that talk, but there's a question. So, yeah, you can think of easy questions that are easy. Okay, just ask them. Right, where are we? We're, consider a champion. Okay, I'm not sure if you know what a champion is?
What a champion is, what you want to convey.
Okay, well, I think a champion is someone that you know in your institute that would just be on your side. If anything ever comes up, they would say, Liz would do that. She would be really good for that. Yeah, I think Liz. Have you thought about Liz? Someone that you trust, definitely trust them, and you just go and speak to them. You say, I really want to promote my career. I wonder if you're ever in the position where you need to suggest someone, you would consider me. Okay, you phrase it carefully, but it can really help, okay? Champion?
It's good because you are short in time.
You can get a word in edgeways around this. Expand academic citizenship. Do you want to do this one?
Sure.
Go.
So academic citizenship is more senior scientists or professors, and you can ask them to help with the review process. I mean, peer review is fundamentally how science happens and how science gets published. Always consider that. And as you're doing that peer review, it's kind of easy to fall into that practice of criticizing the work that somebody else has done. But over the years, I've actually come to view the peer review process as remote mentorship, right? So I have a perspective on a particular topic, and the scientists that submitted this paper have a perspective on this particular topic, and it's kind of a disjointed conversation. And if I have thoughts that are valuable and I share them in a constructive way, then we can advance the topic. If I'm too critical or I don't say anything particularly meaningful, then that's not helpful. So it's all about viewing the community of science and how we can move it forward collectively, because that's the most effective way to do it.
Sure. I'm going to add a little about not necessarily the academic citizenship, but also expanding that as far as your career development, what we were going to talk about your career a little. The niche you can go from being a plant scientist or somebody in the area of phytotechnology is very broad. And then you are not limited to where you are. Even if you are not physically moving, with this collaboration you are going to build, with the people you contact, you can be in your own institution you are now, but your collaborator can be across the ocean somewhere else. So that way you are building your career, you're just expanding the things you cannot do in your own laboratory, somebody else may be able to do for you.
Great, and then just the last two things on the slide. So is there a society, a student society at your institute? Or if there isn't, could you start one? Could you organize a symposium, invite some of your professors to speak? PAUSE is the Early Career Researcher Society. It's the Postdoc Appreciation Week, happens annually. I started up by the US, well done. We do it in the UK and I know Germany, just bizarre, abstract, different countries. So it can be global, so once you finish your PhD, the next step, a postdoc, then further career is moving that way. The next one is gaining expertise writing proposals. And this is you could do this. Okay, we're just talking about a little travel grant from a society. How many of you have written a grant proposal? Oh, this is good. Get your hand up if it was funded. Hey, these are good odds. These are better than we're getting in our... Yeah, I'd be happy with that from my research proposals. And so you learn how to write something, okay? You've learned the skills. It looks good on your CV, and you got some cash. This is a win-win-win. A lot of societies have small grants for travel, but other small things, maybe you want to do a little bit of outreach with a local school. You get money to do that. That is a lovely thing to do, and it gets you experience. It's a good thing to do. Yeah, have fun with that.
I would just add one thing. Just be persistent. I mean, writing grants is not easy. I've always say that there's only two things that you can fail fail at 6 out of 10 times and still be considered successful. One is batting in baseball, and the other is grant writing. So look, it's a process. I think I've only ever written 1 or 2 grants that were funded on the first time. So you take the feedback that you get, you do a little more preliminary research, and you go again, and you go again. That's just the way it works.
I'll add the same thing. Definitely writing more. Be in touch with people, ask them for sample. Because if it's something you're starting from scratch, you don't have any idea, am I writing good? Am I okay? What level am I? You can have sample proposal, or you can reach out to somebody else has done similar work, ask them for their opinion, that maybe they can comment. So you want to also be mindful about the timing, to give yourself time to be prepared for your proposal.
Okay, we better crack on, or on will have me chopped, sliced, and eaten. The science, okay, this is why we're all here. But not all science is good. Question all the time your experiments. Why are you doing it? Because your supervisor said so? Does it make sense? They're telling you to add this muck into that tube and stir it. Is that good? It could be. Just have a think yourself. Maybe ask other people. Say, this is what I'm doing. Do you think this is a good idea? Okay. Read existing literature. Do not repeat stuff that has been published, because people do. I get manuscripts and I see work that has been done 20 years ago and several times since then. Exactly the same species, the same metal. I'm really angry because it's difficult for me to get funding. And when I have funding, I go, I'm going to do this. It's brilliant. I'm really excited. But I don't get the funding, so it doesn't happen. And then they got the funding, and you did that? Such a waste. I'm gutted. And I send it back as well. You're not getting that published. So what a waste of time that was. Think where your outputs will be targeted when you're doing your experiments. What journal is this going to go in? What are those graphs going to look like? Okay, how is that all going to fit together? And we've had some brilliant science at this conference, a lot of excellent science I've really enjoyed. But don't believe everything you hear. Just because someone is eminent and pompous and everybody claps and they go through lots of slides, has it been done before? Did it make sense? Really? Just question it. I'd like to think about that one. Okay. Anything to add?
Yeah, that goes to ethic. I wanted to emphasize on the ethic of the work. You question, and also you learn from other people. If you, for example, you look at the manuscript, you look at the graphs, you see there is no error bars there. The question, why there are no error bars? Did they have enough replicates to create the bar that they can include error bars? Maybe they didn't. Maybe it's a single data point, or maybe 2. That also brings us to this. When you are working, I know we are all short in time, we are short in budget, but we are creating knowledge. We are doing the work that impacts the next generation, impacts us and next generation. It has meaning, it has value. So I want all of us sit back when we are looking at our data. Don't feel the pressure, I'm on the deadline, I should give this to my professor, I need to do this to graduate, and then, okay, that's good enough, send it, go. Let's not do it and think about the consequences and morality of the work we do. If I have only 3 replicates and one of my replicate is the outlier, I'm done. So I'm short in my time. I started maybe also a little late, even if I'm on time. With 3 replicates, you get basically nothing, and that doesn't have much value. To Liz's point, when you see papers like this, it is questionable. It puts pressure also on the editors and reviewers that, okay, I want to support how the knowledge goes forward, but I don't see values in this. So that's the ethic part. I want we all think about this as a society, as the scientists.
Okay, thank you. Great, Azam. I want to bring Jason in to talk about stats, because I remember he, I think, picked me up on one of my manuscripts for poor statistical evaluation. The importance of statistics in your science, rather than saying something probably different, this might mean, what do you say instead?
I don't remember.
I'll let you on.
I mean, my advice, maybe this is it. My advisor used to say, if something is not statistically different, then it's not different. So the statistical analysis has to be there. Without that, you can talk about trends in a paper, but you have to be clear on what's really different and what's not. And that's where experimental design comes in. I mean, and maybe back to this original point where we said, question your science, question your experiments. And if it doesn't make sense to you, you should not be afraid to go to your advisor and say, I'm not quite sure why I'm doing this. What's the big picture here? Because, back to that mentorship thing, they're supposed to be mentoring you. And if you disagree with your experiment, you don't have to walk in and say, this is crap, why are we doing this? You can walk in and say, well, what if we added this treatment, or what if we did this expanded concentration range? How you frame the conversation will make a big difference.
Training needs. Do you want to pick this up? It's a pretty quick one.
Sure, what's that? You started, I think.
All right. What training needs will you need in the future? Then we've got Harry Potter looking into the crystal thing. What's that called? Globe thing.
I'm updated on it.
So now is a really good opportunity to get training, not just your PhD lab, how to run a gel, but presentation skills, time management, programming. There's a lot of bioinformatics coming your way. You guys are going to be sequencing everything on the planet. Do you know how to use Python or R? Maybe you can do online training. These are really easy to use programs. You should be fluent in them. So think about how you're going to access that training, because as students, you should have access to those kind of things quite readily. As you get further through your career, it gets harder to pick some of these things up, and you should already you're expected to have some of these things already. So pick it up now while you can, because you're going to need it in the future, okay?
Yeah, and just to add one point, you can have the best data in the world, and if you can't present it either in written form or in oral form, nobody's going to get it. So, I mean, it's really important to be able to express your data, what it says.
Yeah, one more adding on the presentation skills. I mean, overall the skills you are going to have. One is, don't expect your advisor knows everything. For example, Python or R. We are saying you need, as you move forward. Yes, it's great if my advisor knows that, but maybe they don't. So you are responsible to make sure you are a good fit for the era you are. You don't want to be rusty. You don't want to be outdated. That's one thing. The other thing, we have soft skill, hard skill. Look at the job description. A lot of the soft skills are, for example, critical thinking. That brings to the first question. So if you are able to think about the presentation is delivered, you can think critically. Obviously you can, but then be able to speak up, being able to not be afraid hearing your voice. That's another soft skill. I'll leave that here, but look at the list of soft skills, hard skills.
I hate hearing my voice. Okay, hands up if you think that Azam, Jason, and I are friendly and approachable. It's a bit of a leading question, I'll admit. Oh, thank you, guys. Then I'm sure you will not hesitate to ask any questions from us now while you have the opportunity. So as soon as we step out there, we're going to be busy talking with other people as well. You've got a great opportunity now to ask any questions about our career or advice or anything that you want. Don't leave me hanging. Remember what I said.
While you are thinking, let me add that. How many of you actually went and networked? Networking is very important. When you go to societies, you meet people. I know so many people, they built their next step in their life. They found somebody to sponsor them. Right? I did that too. That's how I got my postdoc done. And so many other people, their career and life is by networking. How many of you did that here? Hey.
Come on, Isabel gave a fantastic talk about LinkedIn. He's not on LinkedIn. Yeah, better keep your hand down and quickly link in now. Okay, thank you, Isabel. That was very, very good talk. I enjoyed that. So, come on, someone's got a question for Liz, even if it's what's my favourite colour. Come on, Jason, what's your favourite colour?
Blue.
Okay, we can waste our time like this, or you could ask good questions. Jay.
I have actual question.
Hey, thank
you. Let's say we have a stage fright and we get completely blocked when we are giving the presentation. What would be a practical advice for it? How to make our presentation, like oral talk, more engaging? Okay. And how to battle that stage fright that I think majority of the people have.
Yeah, it's terrifying, isn't it? Really terrifying. So one thing I think helps is to have on your first, you have your title slide, and then the next slide in, you have text that you read. You don't have to worry about going, it's there, just read it out. Read it out, and then you will calm down. and then look at people in the audience, and you see that they're individuals and that they're humans, and they're not a big, giant dinosaur or monster, but that you're all individuals and you're all friendly, and none of you really want me to fail.
Graduate advisory at a large group, and we had to do weekly presentations.
Practicing is definitely important, and then if you have opportunity to enroll into classes to learn, have somebody to train you, that's great. If you do not, at least have your peers, people sitting, and then you can interact with them. And one more thing to your question, when you are on the stage, being aware that it is intimidating. It is one of the most fearful things to do. If you read about the whole public speaking skills, it is known that speaking in front of the audience is difficult. Having the microphone is another difficult thing because it echoes. It's not comfortable things to do. So giving ourselves credit, being able to do this, that's important. All of you were selected to present, whether it was here or as a poster. Excellent job. See how valuable your work was that was selected and you were chosen to present here. Format doesn't matter, was it orally or as a poster. And look at everybody else that presents. So your work was really valuable. Having that confidence helps you when you are presenting. Now I think we are going to be kicked out.
Any last minute questions? Last chance? Yeah, then stay here, don't leave.
Ma'am, here. Okay, like I have a question. Like when we do research or experiments or like whatever do, there is an urge to produce positive result. Sometimes we do get negative result, but people want us to get positive result all the time. Is it necessary that negative results also get accepted?
Yeah, it's a tricky one.
Can I comment on that? I actually think if you get different results, that's valuable. The point is enough replicate. If you have limited replicate and you are careful, you know, okay, my method is good. I did my literature review. I know I'm doing everything right to check you have a checklist. It's like if you are, let's say if you are a pilot, you have a checklist there. I'm doing all of them correctly. If all of them are valuable, if you have the opportunity even repeat it, so that, I like physical or technical replica, I call them. If I have the resources, I always go 3, meaning I do my biological replicate one set, I get the data. If I'm happy with that, I go another one. Do they match? I'm good. If they don't match, I go with another one. So 3 replicates. You get no significant differences. Right, there was no significant difference. If you get negative result, opposite you got, that's scientific work. My hypothesis is decline. Generate your new hypothesis, go from there. That would be my takeaway.
Yeah, I mean, if science was easy, everybody would do it, right? I mean, one of my regrets is I did not keep my PhD dissertation notebook. And why it's a regret is that notebook describes 123 experiments, the first 101 of which were failures. The last 22 is actually what showed up in my dissertation. But it was a process, and Azam's right. Every experiment informs you on what the next experiment should be.
They still fail. I could finish at the end of January if I just did the experiments that worked. You know, I could just take the rest of the year off. But it doesn't get any better. Okay. Yeah, they're coming in. Come on in.
Yeah, I just wanted to know, at one point we won't be getting results, but then we are getting moderate results, and we just want to keep going on, but then we don't have time. So how to know when to stop a research and move on, or to how to divert it?
You get really good at your PhD. Your supervisor wants to keep you longer. You know what you're doing. It's starting to really, really perk up. You've got to call it in. If you just need experiments for publication, then maybe consider bagging that. But you've got to draw a line somewhere. You've got to think of your postdoc, what's going on with there. So yeah, just have a think.
Yeah, and recognize that, you know, if you're not done with the questions, but you're done with your dissertation, somebody else is going to come in behind you and continue the work. It gets a little easier when you're a professor because you know when to move on when you can't get money anymore. Because if you can't get money, you can't get students or postdocs.
There's also legacy. Your work's going to get handed over, hopefully, to another PhD student. If you make sure that all your work is understandable to them, they know how to access all your files, your lab book, the strains, plasmids, everything in the freezer, fridge, sequences, they can pick it up and they can take it further, which means you get another publication with your name on as well, and everybody's happy. So make sure the legacy is good that you leave when you finish your PhD. Okay, I think we're probably out of time. We'll have my
I just add this. Do a good job and note when you are writing your lab notebook, make sure everything is readable, not for yourself, for the next person. We have this rule, you are not erasing. You can cross, you are not erasing. Next person does not repeat the same mistake.
Okay, thank you.
You stay here, because you all three are moderator for the Phytotechnology Society as well. You two are the vice presidents and no, you are.
I'm presenting the society. Right?
Yeah, representing society.
Yeah.
So, I hope the students had a wonderful session with the three excellent researchers representing the government and academia. Now, the next session is the International Phytotechnology Society meeting, and this is open to all of you, all the participants. It's not that's only for the IPS members. It's open to all, so that you can know what the society is, and you can decide. You can join the society as a member, and you can also use all the resources the society has for all the means publication, access to publication, International Journal of Phytoremediation, etc. They will explain those, and this is 20 minutes, and you can just ask. These are the three. Jason is the past president of the International Society for Phytotechnology, current executive vice president Dr. Liz Rylott, and Dr. Ajam Noori is the vice president. So they are representing the society. Barbara is not here, but they are the leader of the society. So you can ask. They will explain the society. You have some slides. Yeah. So they will explain what the society is and what the objectives and what missions of the society is, and then you can ask questions. Yeah. Thank you.
You're going to close it? Yeah. I just close it and just close it. Sure. And go to session E.
Session A.
Here, towards your right.
Yep.
Yeah. Session E, then you will find your. Can we put chairs for you for the other people? Can we put chairs? Those people are standing.
Sure. I think perhaps.
Chairs.
Where am I?
Yes.
Is it on? See, this is
okay, perfect. All right, so we go to business. Elena is here. Call the meeting open. Official minutes. Yes. Okay. All right. Thank you. So the introduction to the society was actually done on the first day. So I'm going to go brief on that, and this please, Jason, comment if you think something is missed. We can go quickly. So we'll talk about the society overall, overview of the society, who are the members now, and then the businesses we have, and the meeting closure. So the society, IPS, is non-profit organization. It's a worldwide professional society engaged in the science and application of using plants to deal with environmental problems. So that non-profit is the important thing to consider. We are all here working voluntarily because there's something we do care about. The mission is stated here. I'm not going to bore you, read the whole thing for you. The whole intention is educating, doing research, and working towards the common goal that's cleaning our environment using all sorts of techniques that we are aware of, or we can innovate and be innovative and then adapt them. We come to the board, the executive members. Liz presented them on the first day. Barb Sadly, she's not here this time with this conference, but the actual slides, she provided all these slides for us. And the board of directors. Then we have the governance, the term limits listed here for everybody. Executive IPS committees, that's where I'm going to talk a little more. So IPS committees, we have the executives, partnership and growth, benefits, outreach, education and standard. So executive is doing the job, excellent. Partnership and growth, we are working on it. So I mentioned what's going on. I was asked earlier this year, Barb reached out and asked me if I'm willing to chair this membership committee. I said, sure, we'll do that. I asked for help. Martha stood up. She joined. We have Rennie, what's her last name? Renier Stoops, who helps with organizing the website. And then we have David Saw, also Berry, joining us. We met every other month during last year discussing options to grow the society. Through that process, we reached out to members who did not renew their members, so they are past members. We went back to the last 7 years, and almost 7 years, and we reached out to them. Martha was a great help, sending email to people and saying, you forgot to renew your membership. Please come back, renew. Unfortunately, we didn't get many responses. We had only one person who did renew. So that's the part that I'm going to talk a few more minutes about that after this slide, saying why we need a membership. The benefits, as far as the benefits that having the society and the benefit committee is working towards, the conference, excellent, done a great job. Again, shout out to Joss, and all the people behind the scene that did the work. As far as the scholarship, one of the aim of the society was, we would like to do more. We would like to produce more. It's working, but can be better. Maybe that idea of networking and working toward the common goal. The research opportunities, we think we are not providing that enough. We are not this society. Everybody is doing their excellent job in their own laboratory or collaboration with others, but it's not coming through the society, or we are not reaching out from the society. That's something can be done. We do have the newsletter. If you are not receiving, please reach out. We'll make sure Rennie helps you to receive the newsletter. Education and standards, there are some resources available. This is something we want to build more on that towards the membership benefit. So here's come the slide we received from our treasurer, Matt. How much do we have on the account, on the PayPal and bank? So on PayPal, we have $934.58. On bank, we have $6,971.14. That's not enough. That's not much. We can do more, but this is okay. The part here, it doesn't work. Okay. The part that we need to work way more on that, so maybe it is part of, I see from the person who is asked to do, is the membership. Our membership grows when we have meetings. Every year when we do have the meeting, people see that, if I go register through the membership, I'm given $150 or $160 discount, I go become a member. So that way the society, in the bank goes higher, and then we are going to have more membership. Then you may ask, why should I pay? What is the benefit of that for me? We have the list of the benefit you receive. Again, I want to bring your attention back to that non-profit things. It is not like by membership you are enrolling, anybody is receiving monetary compensation. It is just for the society to function, being able to do more of this kind of conferences. So if you feel you got one good thing from this society, so that would be a way to look at this, that maybe consider membership. We do have the program to give discounted membership for certain societies, so a lot of students here will be eligible for that program. So the numbers we see here, we are 39, which is down from last year for the full members, and then we have the students on. So the Elections, next election is 2026, so the members are selected 2 years ago, so will be ASL as was on the couple of slides before. The benefits I listed here, that's what was sent out to people. When you get the membership to the society, you get access to the journal. Jason will talk about the journal in a few. You get the digital access. A lot of students do have access through their universities. That's great. But if you don't, this is one way to have access, free access to the journal. Registration we mentioned, subscription to the IPS newsletter. What is the benefit of that? Let's say you met one of the people here, you want to reach out to them, look at the directory, you see their name here in their email and the work they do. You didn't meet that person here. You go to the IPS website. You see the work people are doing. Again, that's how I found Lee Newman. That's how I went to her laboratory. I found the work she's doing. I read her papers, and I reached out to her. And then we connected them later with Jason. So that's how you get to know people, and then maybe you cross oceans, and then you change your life. Or contact people, have a different area of expertise. As far as subscription to IPS newsletter, I mentioned you will receive the IPS newsletter. Oops, where did I go? Here. And networking we did mention. We are working towards, so these are the first few options here. These are already in place. They are available for all the members. They receive them. The rest of them, we are working on them. We would like to limit the awards, to provide award and limit those awards to members. We would like to have career development resources available. We have people already volunteer, and they are working toward that. And then phyto training. So David and Barry, they say they do have the resources, and they would like to provide this training for individuals interested. We have been thinking about ways to improve our membership. So here how our membership dues expanded, and then how we can improve them. I'm not going to repeat myself, so I went off the side, just mention some of them. We use the dues for society website, training materials in the discipline provided. Some are there will provide more. And the grants and scholarship, and organizing these conferences every year, hopefully. And here is the businesses we have, so other businesses. 3 questions to consider, and then if you have anything to add, we can add that. Discussing the option of adding chapter to the society, for example, a chapter in India, that's one. Establishing a presidential advisory council, that's another one to consider. Establishing partnership with Root and Shoot. If it's okay, I'm going to briefly mention what is Root and Shoot to have idea to think about it, and then we go back to the last 2. So Root and Shoot, I have this link here. I hope it works if I click. It does. Yes. Okay. Root and Shoot, I reach out to them. These are funded by NSF, National Science Foundation, and the state. Their intention is awareness about plant sciences, and all the work is done by plant scientists. It's like a bigger umbrella, and say organizations and societies are working toward this. Nonprofit societies come, and then we get together. What they expect from us, if we do under that umbrella, is we maintain our own identity as we are, like all these examples, but we do serve as volunteers. 2 members from the society go and serve to help meet the criteria they told NSF they are doing. So there's a long list. We can discuss that if something is interesting. The information about them is on their website, and the societies that are already under Root and Shoot, working with Root and Shoot, are here. So I go back to the slide, considering the time. Looking at the other 2 options, Indian chapter, potentially adding another chapter. We can talk about that now. We can just brainstorm and discuss later. Also, Presidential Advisory Council. Okay. Anything else to add? Okay. So And
Ajam, I'm not sure. I think I was engaged in something else. Did you mention about the reduced rate of membership to Indian in India here or anybody, any country, $75 reduced membership?
Yes, I did mention that, and the fee is reduced for certain countries.
And also, please explain that how that works, because two members that we approved, I sent the email and their board approved, but not able to pay because the amount, $75 amount, it was not accepted online. So explain that how the reduced membership can work and how we can pay for that. That was an issue that brought to me here in this conference.
Okay, that's something to ask. I'll reach out to Matt and Rennie. Supposedly it should work, but I'll ask Matt and Rennie too about that.
Thank you.
Thank you. All right, so the first question, anybody has any ideas? Also maybe board members or other people? Should we consider adding the chapter to the society? Any ideas on that?
No, one of the good things, I think, it's to have the conference in North America and in Europe especially, and rotate after that even to have between Europe and Asia. That's the way, and that's in fact the work of the member, to disseminate the information, and especially to the PhD student or the postdoc, and to motivate them, and pass the message that in there, when they are bidding for proposal for a project, to include the money for dissemination and attendance to the conference, and to put it on the schedule in the project that you will present your work or your outcome from the project in a conference, and especially at the EPS. That's what they are missing, because they start their PhD, but suddenly they don't. Generally, they are running their grant with side by side with a project, and in the middle of their PhD, they discover that they have to disseminate. But at the beginning, the money was not putting in the project, and it is in fact also the work of the mentor to think about that and to include the money for the traveling of the PhD student and attendance to a conference or many conferences as much as possible, and to include that in the bid when you bid.
Yeah.
Thank you.
So the chapters, I think that idea was brought by Professor Josh Puthur to have like if we can explore the Indian chapter of the IPS here. But I think that's an excellent idea in my view. As you all know, that I've been involved in several other societies as well, ASPB and others, and ASCSESA. So they have those regional chapters as well as country-specific chapters as well. And I think having a kind of either regional or country-specific chapters, that can help the society to increase the membership, and also the members can benefit from the local chapters. They can have their yearly local meetings and all those, and they can discuss the issues. And then these members can come to the society meetings whenever the meetings are happening. That's I think that's a positive sign for that. Thank you.
So we are, thank you. Our intention isn't like voting now, collecting votes. It was just receiving feedback and then moving forward with our meetings next time. The second option, establishing a presidential advisory council. Any comment on that?
One thing that could be also very attractive for the EPS conference, it is to maintain some keynote lecture that somebody can address really like we do for the microplastic. I think that in this case, the mentor recognizes that it will be a very good continuing information for the PhD student, even for them, even for me. And the keynote lecture, we just have, let's say, one really in this conference. I think it's very important for each topic. I mean, the POPs, nanoparticles. You did it, Jason, more or less, yes. POPs, microplastic, constructed wetland. We miss the people dealing with constructed wetland, for example, here, and it is very, very important point. Another point, so we connect lectures that can help. Another point, another idea, partnership with Root and Shoot. I think we need to have a stronger links with Société of Quebec Phytotechnology, Society of Quebec Phytotechnology, because finally we are doing the same. And I think we could co-organize sometime, like we did in Montreal. We could co-organize a little bit. And many people attending to the Quebec Society of Phytotechnology, in fact, they could be, maybe not in the EPS, but some are, like Michel Labrec, he was included. But many of the could be, let's say, in a side-by-side or co-partnership. And of course, I noticed we have two consultants, two companies from the state. But I remember when we were in Syracuse or especially in Syracuse, so many companies from the US were there. So we need to advertise also the company and give them space and say, no, we are not only addressing science at the level of the frontier of the science, but we are also welcoming consultants and company to demonstrate where the phytotechnology are really applied. What are you really doing? I remember yesterday we are in the POP session. We have a consultant presenting the work at a TC contaminated site. It was really interesting to see in practice how they manage and what's the problem they encounter. And I think we miss a little bit this companion.
Thank you for the feedback.
Okay. Yeah, thank you, Michelle. I think that's really important, and we will bring that up to the next online meeting for the IPS. We will have a higher number of members present online, and we can make a big decision there. We also have to decide for the next conference, and in the past, on occasion, we have announced the venue for the next conference, but this year we have a number of different proposals, and we're still debating the costs and the pros and cons of where the next IPS meeting will be held. So you will be hearing about that in the next few months, voting for the members on who it should be, and then more information on that conference when it will happen. So I guess watch this space is the answer there. So next thing.
Yeah, if there is no other comments or points to discuss, I think we are done. Call it closed, Elena.
Closed.
Thank you. Thank you. All right, now we have the awards. Yeah.
Thank you very much.
Jason has a journal.
Okay.
Oh, right. So I think this will just take a second. I think many of you are aware that the Society does have a journal associated with it, the International Journal of Phytoremediation. IJP actually predates the Society. The journal was founded in 1999. And what we have historically done, with the exception of last year's meeting, is we will have a special issue of the journal from research presented here at the Society. So you will see an email on that coming out in the future. The journal, the impact factor is 3.4. We get between 460 and 550 submissions a year. This year is going to be 550. And our acceptance rate's about 35%. So it's a good journal. And you'll see instructions in your email inbox about how to submit. Yeah, and the papers that will be submitted, they'll go through the regular peer review process of the journal. They'll just be collected into one issue, those that survive peer review, which hopefully will be more than 35%. So.
Well, we would like to thank Jason and Liz and Ajam for excellent overview of the society as well as the journal. Thank you very much for your contribution and your leadership. Thank you.
Yeah, yeah.
Yeah, that will be the first she will adjust. You got this one? First you will adjust. Then you get you announce and you make a 2 minutes in general you can talk. Yeah, you can take.
Thank you all. So now let us begin the valedictory ceremony. We have with us the chief guest of the day, Professor E.K. Satish, Registrar, University of Calicut, and I invite Sir to address the session.
Dr. Liz Roylott, Dr. Jos Putur, Dr. Om Prakash Dhankar, and other dignitaries, students, delegates, researchers, ladies and gentlemen. It gives me immense pleasure to stand before you to address this august gathering, which is the 18th edition of the IPC and first in India on a very important topic, Phytotechnology conference and phytotechnologies for sustainable environment and food safety. Actually, we have started discussing about sustainability on a summit held in Rio de Janeiro in 1992, where the world joined together to convene a conference which is called the Earth Summit. As a result, we have globally developed 17 sustainable goals. Since then, cutting across disciplines, we have been discussing on this, on different aspects of sustainability. We have seen many conferences, many ministerial meetings, altogether different forms. Some of the forms are below the sea and some of them are on Everest. The ministerial meetings conducted by Nepal was in and Mount Everest. Maldives conducted its ministerial meeting in below the earth. Actually, so many conferences across the disciplines. Some of them are sustainable consumption, some of them are sustainable tourism and whatnot. And India as a country is perhaps one of the leading country in bringing this phytotechnology, particularly in producing alternative energies and also to protect the earth from possible contamination. I am very happy that this conference is being attended by 19 participants from across the globe and 52 participants from outside the state of India, of Kerala, or 60 participants or 220 participants from India and also outside India and also from Kerala. This conference, I believe, was one of the successful one, and you have many takeaways from this conference for making the future life some sustainable one to all of us. I hope that you will be coming out with excellent contributions in the form of some seminar papers or in the form of books, which will be a great contribution as an outcome of this conference. I sincerely wish all the best to the participants and hope that you can enjoy the ambience of this university and also the particular part of India, and can have an excellent networking with academicians and other researchers. For we have only one Earth, and that have to be maintained and have to be handed over to the next generation or next to our kids without much problems. I congratulate the Department of Botany and also the people behind this conference and the idea that you have transferred here, and which will be triggering so many conferences associated with it. I wish you all the best. Thank you very much.
Thank you, sir, for your motivating words. Next, I would like to invite Dr. Martha Marbiroli and Dr. Elena Mastry to announce the award winner for Distinguished Service Award.
One moment. Okay, just wait for your awards a little longer. I just want to summarize the conference. I know that Barb wanted to be here so much. She tried so hard. She made a lot of personal efforts to be here, but it was not to be. So I've been emailing her, and several of the others have as well. We've been sending her lots of pictures and updates. She's following it avidly, and she's absolutely gutted not to be here. But what a thing that she missed. I feel so sorry for her. 3 days, free phyto days, 3 fantastic phyto days. What amazing science we've had. I have really, really enjoyed this. The science, the POPs, the metals, the wetlands, emerging contaminants. How many sessions was it? 8 sessions. It's been an amazing experience. I have hugely enjoyed the science. The location, oh my goodness, Kerala is beautiful. This campus, you are so fortunate. I could stay. I think I'll be in trouble. I'll probably get deported, but I would love to stay. It is beautiful. Your climate is lovely. In the UK, it is cold and wet and raining, and I'm not so keen to go back. I know when the plane touches down in Heathrow, the rain will hit the windscreens, and that will be it. I will sigh. The science, the location, the food. The food, I'm a little bit fatter than when I arrived. Thank you very much. The food is delicious. You must not lose part of the food. And the dancing, oh my word, how much dancing have I seen? Wow. I've seen dancing. I believe last night I may have married an elephant. I'm not sure if that's true. And we've seen children very nearly cut in half, but thankfully just cucumbers. Wow. This whole event has been put together on the back of many people, okay? The people who put the carpets down, the cooks who filled my tummy, the people who put the poster boards up, everybody, the AV people. Hold your hand, we'll do some clapping in a minute. The AV, thank you very much, guys. Oh, go on, clap the AV, thank you. People who've been keeping us all to time. So I know there are many student helpers, a lot of them from JOS and OM, who've been helping, and we've seen them running between sessions. We've seen them announcing stuff, running with microphones and all sorts. So a round of applause for all the myriad of helpers that we know have been doing really hard work. Thank you. Then I know that OM and JOS have lost a lot of sleep over this conference, a lot of personal time. I've broken sweats numerous times online on Zoom. And Barb, Elena and Marta for organising the judging of the posters and the presentations, and all of you for attending. But I think we really need to give a big round of applause, a huge, bring the roof down please for JOS and OM, come on. I hope that you will go forward from this conference with knowledge, confidence, and impact. I hope that you all have been changed in a positive way, and this is something that you will remember in a really good way, I hope. And it's something that you will go on to learn more and pass down. And I really, really hope to see you again at future conferences. If you see me, come say hi. I'm always up for talking. I might even shut up so that you can get a word in. Okay. Thank you so much, everybody. And now, the awards. He was talking. Yeah.
Thank you, ma'am. So I would like to invite Dr. Elena Mastry to announce the award winners for Distinguished Service Award and Studer Poster and Presentation Award. And also I would like to invite Dr. Riz Railot and Dr. Josti Putur to present the awards to the winner.
So where are you going to do the presentation?
Okay, so I will do the posters.
Is it understandable?
I'll do the posters, you do the platform?
According to one, I read the name
Platform, okay. Do you want to read the name?
The platform first?
First the platform,
Sure.
That is arranged.
Okay.
You want platform?
Because they arrange the posters first.
Let's decide if it's poster or platform.
Poster.
Poster. Okay. So let's start with third place poster. Correct. So I'm sorry for the pronunciation. The third place poster goes to Josfat Kiunga.
We can announce what all it will have. It will have a medal, certificate, a gift.
The prize are a medal, a certificate, a gift, and money. Stand, okay, in front of the camera. The cash prize, we remember, it's $50. And the special gift, we don't know what it is. What's the special gift? A surprise. Ah, spice. Wow. Ready for Are you ready for second place? Second place, Sandra PV. She probably will not need the spices. The money in this case is $100. And the spices, probably she knows them. Wow. So, getting ready for the first place. We have to clear the floor first, okay? The first place for poster goes to Atish Kumar Behera. Okay, these are $200. And the spices. Very well. So now you want to take a picture all together?
No, all six.
Ah, all six at the end, okay. Okay, so we move to platform. Yeah. Platform. Third place for the platform presentation, Erin Lincoln. Again, we have the medal, the certificate, and $50. And the spices. Second platform, Ria Johnson. $100. Very well. Now the first platform is the most difficult name to pronounce, Pietro Peroni. Congratulations. $200 and the spices. Okay. You don't need
Yeah, go.
Marta, come with for the picture.
One more.
We see it.
Okay, everybody, there's just 2 more prizes left to give, and they are the Distinguished Service Prizes. These are for people who have gone beyond, above and beyond, made that extra effort, gone that extra mile, and boy, have they gone that extra mile. They've gone a long way, they've worked incredibly hard, and I think we all know exactly who they are. It is, of course, Jos and Om. Thank you very much. This one to you? Yeah. Can you shuffle that way? I've got the lectern. Well done, Josh. It's been amazing. Really, really pleased. Thank you.
Thank you all. Now moving on to the end of the conference, let us hear the closing remarks from Dr. Om Prakash Thankar and Dr. Jos Tiputtur. Sir, please.
Wow. First, thank you very much, the Society, for the award. I was not expecting, honestly, because this is the Distinguished Service Award, not for this 3 days conference. It's a career-long award for the service to the Society. So thank you very much. Appreciate that. And now, I'm a bit emotional too now after 3 days. And I don't know what to say, but I hope you had an excellent time here and wonderful conversation and networking and great science. And I hope you are going home with all these deep memories and reflecting on the 3 days of conference here and the friendship and all those networking that you have made. And I hope I hope you had a wonderful time. As a co-chair and organizer, I'm no one to judge how successful the conference was. It's to you to judge that. So me and Jos are just happy to hear your comments later on. But there are so many things behind this. And I have my long list of thanks to many people here. First thing is, before I go on the others, first thing are the presenters and participants. You all did an excellent job, presented excellent science, and all this camaraderie and networking. Thank you very much. Second is the session chairs. We are deeply thankful to you all for the effectively managing the sessions on time, and if not only time, but also probing the students to ask questions, like what Liz was doing, I saw that. So probing the students to ask more questions, and that's basically how you should do that. Students should come up, and when you think about, you may have a question in mind, but sometimes when I was at your age, when I was sitting in the classroom or in the conferences, I had question in mind, but when I start feeling like, should I ask, my heart start pumping. And I felt like, I don't know. But there's no stupid questions you should ask, and people will respond to your questions. So just get the fear out of that, okay? It will come automatically, slowly. So and then third is the society. Thanks to the society for selecting this University of Calicut for this venue for the 18th IPC, and interesting in having the faith in me and Jos to run this conference effectively here. So we are very thankful to the society and their leadership for putting this conference here. Thank you. And also providing the resources too. Hospitality. Can't say, it's whatever thanks you put for the hospitality is, the thanks is a very small word for that. So in that way, I really, I hope you had enjoyed all these mouth-watering cuisines of the best cuisines of Kerala and the hospitality and all these students that how they help and all this effort they put into that one. Thank you very much. Also, I would like to convey on behalf of the IPC 18, I would like to convey the sincere thanks to the Vice Chancellor and the Registrar of the University of Calicut to provide the facility, hosting us here, and all the resources they provided and leadership they provided. We are very thankful to you here. Thank you. Okay. And of course, yes, mesmerizing cultural programs. And I'm sure Liz will never forget that moment yesterday that she had yesterday. I captured that on the photographs, and I was planning to put on the slides, but I thought that will be maybe too much. She will be blushing. Actually, this was my long-term wish. I've been associated with the society for a long time. It was, I think Jason and Liz and everybody knows that it was my long-term wish to bring this society, the IPC, to India. I've been working hard for many, many years, and Michelle know that. He was commenting during the lunchtime. So it was fulfilled, and my whole objective was to bringing this conference here to India, engaging the Indian scientists, and the Indian students and researchers to all these world leaders in the phytotechnologies, so that you can develop collaborations. You need to know that science never grows in silos. You need to collaborate. You need to make teams, and then have basically the kind of like the tackling the global issues from all angles. One cannot solve all the problems. So collectively we can solve the problems. So collaboration is the key, and I hope you have developed several collaborations and networking from this conference. And then, last but not least, I cannot thank enough two teams. I divide this the host here in two teams. Team Calicut, all the people from the plumbers to all the decorations and all these people putting all this IT and everything together. That's thanks to you for all these efforts. We are highly, highly thankful to you. Second team is Team Josh. Team Josh is all these people from the Botany department who put several months, I think maybe almost a year we started that, and then as the time come closer, our meetings from the monthly to the weekly to the daily, 2 hours, 3 hours, phone calls or the Zoom meetings. It was, and then the local team here, they manage everything so efficiently, and they spend so much time. They are basically, as Dr. Bhaushan Singh said, that they are the real unsung hero behind the success of this conference. Thank you to you all. So I hope this conference has generated a lots of interest among students to pursue their research and career in the phytotechnologies, for the nature-based solutions to solve the global issues of the pressing needs of the environmental challenges of the society. And I hope that you will pursue your careers in that. There is plenty of opportunities and career options for this in this area. And all the participants, I hope you are going with a lot of good memories about the food, about the culture, and I hope you will have a good time, rest of the time here when you are here. Anybody who is going for tours, please enjoy that. And thank you to all for a wonderful conference, and have a great evening, and have safe travel. Thank you very much.
Respected dignitaries, my dear students, good afternoon to all. 2 days before we had the curtain raiser of IPC 18, and today it is curtain down. So in these 3 days, what I felt is that we all have become a family like. We were enjoying, I think by the second and third day, we were all very close friends. Even I saw that on the first day itself, some participants were dancing with our students in the garden. And they were enjoying. And other side was the serious part, that the sessions were excellent. And all were sharing that although they expected not such good talks to happen, but very good talks have been taken place here, and it was really academically rich. A lot of gaining of knowledge has taken place, and that is basically what we expect from a conference of this standard. And in the beginning, in the opening remarks, I told you that the main aim of conducting this conference in university was that we wanted our students to get an exposure to international as well as national stalwarts in this area of phytotechnology. And I think we could get a good number of stalwarts in the area of phytotechnology from within India and from abroad. And they have really interacted with them, and that is the exposure what we wanted. And I think our aim is achieved with these 3 days. Now, just one thing before I forget. The certificates, I made an announcement in between. The certificates all are available online. You have to just scan the QR code on your ID card and it will be downloaded. Today morning onwards, it is made available on this QR code. Directly it will go to your certificate. So if it is or a presentation, it will be that, or if it is sharing, you will have, if you have done both, you will get both of the certificates. So that is one thing. And another thing, if you have missed any of the talks, all the sessions are made available in the YouTube channel IPC 18. And that has been shared. If anyone has not got the same, I can share to them. And when you are free, you can listen to the talks, if at all, because parallel sessions are going on. I'm sure some of you must have missed some of the good talks. And you can obviously see that one later also. Now I come to my task. First of all, I thank our registrar. The facility has been provided to us excellently, as already Om has told. The facility, what you see over here, the whole thing was at our disposal. Even the security was telling that we cannot keep it open the whole night. But finally, for the arranging, the previous day, 21st, the hall was kept open for the entire night for us to arrange. And our students and faculty left from this venue around 2 o'clock in the morning to make all the arrangements necessary. And the team which was working behind, they were working all throughout the night to make it final. So that sort of freedom and liberty was given by the management side. And also, I told you, just for this conference, certain amount was sanctioned so that all beautification will be done in and around the Senate. So especially I thank the administration, and the head of the administration is here, our registrar, Professor Sadish. He is one of our colleagues. And I thank you, sir, for coming over this function and gracing this occasion. Now I come to Om. In the opening session, somebody told me, you missed Om. You didn't comment on Om. But I told Om that everything has been reserved. We say that we have fireworks at the end of any program. So we have reserved it for the fireworks. See, as I told you, the history is like this. When he came to for a conference in 2022, he had an idea of bringing IPC to Haryana, his own home university. Hope no Haryanis are here. No, no one is here. So I can talk that plainly. Yeah, you are here. Please don't disclose this. Then at last what happened is that seeing the environment, seeing the condition over here, he said, can you host this conference? Then I said, we have to discuss in the department and take sanction from the university authorities. And that was done in no time. And that is how we sent the letter to IPS saying that we are ready to host. And that is how this has been ready now. And Om, from that moment onwards, Om has been 22. 2 years continuously he was monitoring. In between he came just to see how the things are progressing, to see the venue, finalize the venue. So he has taken a lot of pain in bringing this conference and organizing this conference. As he told, we were conducting online meetings almost earlier month-wise basis, weekly basis, and when the conference was nearing, it was on daily basis also. And in fact, it was his morning hours, but it will be my late hours when I am almost going to the bed, I will get a call from him, WhatsApp call. Just few minutes, but it will end up around 12 o'clock in the night. But I stand on and answer to what all queries he has got. and the thing has been finalized or everything has meticulously planned because of his planning. I thank him specially for your all sorts of support. I am naming not individually.
Actually, thanks to Josh. Because he's the person behind this all. I was just providing my feedback to him.
Now your chance is over now. Now is my chance. Okay, now coming to IPS. I think IPS had some apprehensions in the beginning of organizing this conference. Maybe not because it is India, but it is very far off. Still, some of the members could not attend, not because of the flight ticket and all, but they say that it's a long journey. Long flight. You all know how much time you have taken for the flight and how tiring it is. So they had certain apprehensions, but again, Om was intervened, and finally he convinced them that it will be done very good over here, and that is how the thing has come. And I congratulate, I also thank the IPS for organizing this event over here. Now I come to my department. Of course, I told the department, without the permission of the department and the administration, we cannot go forward. We put up the matter in our department, and wholeheartedly the whole team has supported, and that is how this has become reality today. They said that we can go ahead, and that is, I thank my entire faculty members. Head of the department was here for the opening remarks, today is not here, and I thank the entire faculty, and especially I want to name a core team which has really supported me. They were throughout this process with me. So I want to mention core team, and I also, on the behalf of IPS as well as from the organizing committee, conveners, we discussed in the morning with Om also, that we are giving a special appreciation to this core team of faculty members. So kindly I invite them to come up and receive the appreciation token of love from the IPS as well as the conveners, co-conveners. Please come up, Professor Santosh Nambi, Professor Sunoj Kumar, Dr. Manju, Dr. Pramod, Dr. A.K. Pradeep, please come up to the stage. Lissy, you can honor them with this. These 5 people were my backbone in conducting this program, and I appreciate their efforts. Thank you all. The support given by 2 people will be specially mentioned here, and there is also memento for them. I invite Nidesh, who has given the digital support to us. It was highly efficient. The whole digital services what you have obtained on your phone, your mail, everything has come through his efforts. I appreciate his efforts, and I also invite him to I also invite Aziz, who was the event manager of this whole program. What our arrangements, what you see over here, have the efforts of Aziz. Aziz, please come up to the stage. Yes, please. Now just 3 more minutes, 3-4 minutes, then I'll wind up. So now also I take this opportunity to thank Department of Botany staff, being conducting in a state government university, all files has to be moved, approved, then only we can proceed. Lot of file work has been done. I appreciate the efforts of our office staff under the leadership of the section officer Smita and team. And now I have to specially thank my team. We call it as physiology team. All are sitting here and there. And each one were given a portfolio. If it was one with the task of getting the MEA clearance and MHA clearance, you know how difficult it was. Each of the foreigner would be knowing how difficult it was to get a clearance from our External Affairs Ministry and Home Affairs Ministry. So one was given the portfolio of that, other with the cultural program, other with the venue arrangement, registration, and transportation, accommodation. If I missed it, don't forgive me. So all have been given one task each, and under them, above them, they had a faculty member to control the activities, and together with them, they had all the other colleagues from various sections, various laboratories, and also our PG students and also integrated students, all worked in a team. So I congratulate and also thank each of the students, especially the physiology team and each of the team members, integrated. First PG, second PG, research scholars, and postdocs who all are here. Regarding the cultural program, already Om has told, so I am not repeating it again. Now coming to the physical education department and engineering, they've given a big support to us. We already utilized their entire facility. The director of the physical education department told, treat it as your home. Take it as your home and do whatever you want. That is the freedom was given to the space or utilization. I thank him and also the entire physical education department. Then, of course, our electrical engineering department. In between, we had a AC fault in between. The first day afternoon, you must be remembering it was very hot inside because our ACs has gone down in between. But they worked that night, entire night, and around 9 o'clock they got it rectified. They were supposed to be here till 5, but they rectified it at around 9 o'clock. That is the effort that has been put. So the engineering, electrical, water supply, landscaping, and the garden. These are the staff which has supported a lot to our activities. Now coming to the, not last and least, but this should have been come to the first, the participants. Already Om told, without your participation, rest everything is materialistic. The actual participation of the participants has made this conference a success. You have traveled from far and wide, especially I told more than one day journey you have undertaken, and you have come, you have made this conference successful. Good deliberations has taken place, interactions has taken place. I think a lot of collaborations also will come up after this conference. So I thank each and every participant. I'm not naming each and every one, but every participant for their active participation in this conference. Before I end, I just want to make a small announcement more. You will have a vehicle, the traveler, that big vehicle is available. So whoever wants to go back to their hotels, only one will be available for the entire, because most of them have left and some will be moving in another direction. So there will be only one vehicle and they will be taking to all hotels back. So kindly after the tea, you be near the gate so that easily we can board the vehicle and go back to the hotels. And before I stop, I just want to invite two people. One is Dr. Pawan Agrawal. In fact, because of shortage of time, because Om told we are short of time, we won't take many remarks, because if the remarks will go on, it will never end. We may have to have another session. But I invite Pawan Agrawal for a minute, because he told me I have to give certain remarks. So I invite Pawan to...
Thanks for the opportunity, Josh. On behalf of all the delegates, I wish to express my heartfelt thanks to you, Om, for organizing this conference. The most impactful event in this whole thing was how respectful, humble, and ever helping with a smile on their face all these students. That will leave a lasting impression that will leave a lasting impression on my mind. And if I ever come to Kerala, that will bring me, said, God, these people are so humble, so respectful. What a bunch of people they are. And mind you, most of them are very intellectually, highly, highly capable. Thank you, everyone. And Josh, you have done a tremendous job. Believe me, we are leaving with a heart full of appreciation for you all. Thank you.
Now, not exactly as a part of this event, but it's a small event for a minute to occur. That is regarding International Society of Environmental Botanists, which is a scientific member of International Union of Biological Sciences. And Dr. R.D. Tribadi, secretary of this organization, is here. He has a surprise, and for a minute I invite you for that surprise.
So, good evening, everybody. I come from the National Botanical Research Institute, Lucknow, where there is a society, International Society of Environmental Botanists, which is a scientific member of IUBS. And we had a lot of friends in the United States and many parts of the life members and society, and they have been. So, every 4 years we have a conference. So, I wish to invite many of you. It is the October 2025. So there is ample time. So, and there is one thing that I had a very, I really enjoyed, and I felt it was more enjoying when I came to here. It was beyond my expectation. So listening to Jason and Boshanjing, Om, and so many people I cannot name. So really it was a scientific fragrance. Beside the cultural fragrance was already there. So, and Professor Josh and Professor Dhankar, I know them from quite some time. And again, they have done the tremendous job. And Registrar Sir has already told. I had one more surprise about Om, that he's a scientific all-rounder kind of person. So, in fact, Professor Rana Pratap Singh is here. So he was in the selection committee, and in 2022 we could select him for the fellowship of the International Society of Environmental Botanists. And he could not come because of his engagements and Corona and so many things. So I think it was a good occasion to bring the certificate and the plaque. So I would I invite Professor R.P. Singh, who was in the selection committee, to give him the award. And it was an additional admiration for Om Prakash Dhankar ji. Professor Josh, you can understand. Liz, you can. Yeah. Professor Dhankar.
It was surprising.
Yeah.
It's embarrassing to me. Because this is a different society and this is a different occasion.
We had the permission from Jason and many others, so I was a bit of part. Thank you. And these fellowships are advertised, so I will share the advertisement to all the members of the IPS, so that they can apply for the nominated for the fellowships. Thank you.
Thank you, sir. Now, let us all rise for the national anthem.
The vehicle will start at 4:15 from in front of the gate, so kindly be there. Those.
No, we have to say tell them now.
Tell them now.